Kerala, University of Exeter, UK, University of Zululand, South Africa, supported by Ministry of Earth Science, Government of India. A warm welcome to all of you, and today some interesting presentations are waiting ahead. Without further delay, let me invite the session chairs, Dr. Irinjalakuda and Dr. Linto Alapat. Before we begin, let me give you some instructions. Participants are requested to kindly mute yourself and also off your video so that we do not get any interruptions in between. And our feedback link will be posted in Zoom as well as in YouTube. It won't be given in the Telegram. Thank you all for your cooperation and have a great time.
Thank you. Linda, do you want to say some words before I start?
No, you please go ahead, Angela. I think we all had, yeah, please.
Thank you. So welcome everybody. Again, the format will be like yesterday. The speakers will talk for about 20 to 30 minutes. Then there will be time for questions. If you're following us on YouTube, please post your question on the chat. If you're on Zoom, please post it on the chat or raise your hand so I can, I know that you want to ask a question, and then you can maybe switch on your camera and ask the question yourself if you want, or just post it in the chat if you don't want to talk. And without further delay, I'm going to introduce the first speaker of the day. I'm very excited to introduce you to Sakonvan Chauchai. Dr. Chauchai is a lecturer at the Department of Geology in Chulalongkorn University in Bangkok. And she's gonna talk to us about the history and variability of monsoon in Thailand based on lake sediments and speleotherms records. So without any more delay, Dr. Chauchai, please take the floor.
Hello, everyone. Do you hear me? Okay. Good afternoon, everyone. Thank you, Linto and Angela, for your kind introduction. Today, I'm quite a little bit nervous, but I'm very happy to get a chance to share about my research with you. Today I'm going to talk about my research in Thailand, which is about the monsoon, lake sediments, and speleothem record. Let me share with you with the slide. Right now I cannot share the slide. Can you see my slide?
No. Is it possible to allow Dr. Chauchai to share her slides, please?
It's done now. Would you please try?
Okay. Now it's...
Yeah, it's coming.
Okay. Okay, it's worked now. Okay. First of all, I would like to show this figure from the IPCC report in 2018. The upper figure shows projected change in mean temperature of global warming compared to the pre-industrial period. On the left top is 1.5 degrees Celsius, and on the right-hand side, 2 degrees Celsius. For the bottom is a figure of mean precipitations. You can see that about half degree Celsius difference in temperature can lead to changes in precipitation pattern in some area around the world. Especially the changes in monsoon regions caused by temperature gradients between the continent and the oceans. The global monsoon regions shown here in the light gray color in these figures. Here in the red square, show the location of Thailand. Thailand is located in the Asian monsoon system. As precipitation changes, storms, flooding, and drought may occur more often and may become more intense in some areas. In this context, changes in monsoon rainfall are of great concern. Observations can provide a key basic for understanding their long-term variability. However, based on direct measurements of temperature and precipitations, so far I know the longest record is in India. It's go back about 200 years, but it's still not enough. Therefore, paleoclimate dataset is important for a good understanding of long-term climate dynamics to reduce uncertainty and can be also useful to predict future climate change. Lake is one of natural archives to track environmental and climatic change because they store all information available not only from their direct catchments but also from a bigger area. Climatic fluctuations, fires, human activity such as industry, agriculture, and many other factors can be tracked with the use of lake sediment. Scientists work on calling platform. We do calling to retrieve the sets of sediments that will cover the whole history of the lake development. The lake sediment can be analyzed based on physical, chemical, and biological proxy together with radiocarbon dating. These all parameters can contribute to understanding the lake response to environmental change that are related to climate or human activity. You can see very nice biological proxy, diatom, pollen, and some microfossil here. And these figures shows our study site in different parts of Thailand. Today, my talk will focus on the study of Lake Gumphawapi and Lake Pako from northeast Thailand, and also Speleothem from Ban Lai in the western part of Thailand. For the present day climate and monsoon in Thailand, the southwest of Indian summer monsoon brings warm and moist air from the Indian Ocean towards Thailand, causing abundant rainfall over the country between May and October. Between November and February, the northeast winter monsoon brings cold and dry air mass from the Siberian anticyclone over the country, causing dry conditions in the north and precipitation in the south. Tropical cyclone from the east contribute some additional precipitation during the year. In addition, the northward migration of the Intertropical Convergence Zone, ITCZ, coincide with the beginning of the southwest monsoon. It arrived to Thailand in May, moved northward to southern China in June and July, and then it moved southward to northern Thailand again in August. Based on the observation of the present day monsoon in Thailand, we hypothesize that a stronger summer monsoon results in higher precipitation and catchment runoff. This leads to an increased input of terrestrial material to the lake and to higher lake level. A weaker summer monsoon would result in the opposite case, less precipitation, less runoff, and lower water level. And this map shows the map of Gumphawapi and Pako. Gumphawapi is one of the Thailand's biggest freshwater lake, and Pako, which is located about 15 kilometers southwest of Gumphawapi, is a flooded wetland. The distribution between open water is in blue color in the map here, and wetland area in Gumphawapi vary seasonally, but it's nowadays it's controlled by dam in the southern end of the lake. The lake circle here show the calling points for this study in both lakes, called CPTA and called CP4. And we did calling and all the geochemical analysis. This is our result. The upper figure shows lithostratigraphy and geochemical data for sediment CPTA, and the lower figure shows data for CP4. Y-axis, depth and lithostratigraphy, x-axis, geochemical multiproxy. The total organic carbon, or TOC, is the percentage of organic matter, which is related to past lake productivity and preservation of the organic material. The carbon and nitrogen ratio and delta C13 isotopes are used here to identify the source of organic matter. Sulfur isotope values are used as an indicator of the influence of groundwater and changes in groundwater table. For the CPTA, diatom show percentage of carbonic and benthic diatom. And for CP4, the bottom here, is biogenic silica in weight percentage. Is an indicator for siliceous microfossils such as diatom and phytolate. There are two major chains in lithostratigraphic and geochemical data. The first chain is from Gudja K from the bottom, Gudja K sill to peaty Gudja and peat. From low to high organic matter content, from a mix of aquatic and terrestrial organic matter to terrestrial source. Also, distinct changes in sulfur isotope values can be seen. This means that the lake level and the groundwater table decrease. The second change here is indicated by the shift from peaty Gudja and peat to kayi Gudja, or from high to lower organic matter content, from terrestrial organic matter to a mix of aquatic and terrestrial source. This shift imply an increase in lake level, which is also supported by sulfur isotope values and diatom. Okay. For the chronology, also we did a lot of the radiocarbon dating.
Mo, we cannot hear you. You're muted. Your phone is muted for some reason. Now, okay, I think it's okay now.
Can you hear me? Okay. I'm not so sure since when I was muted.
I think you need to start again from this slide.
Okay. This figure shows the chronology. We did lots of radiocarbon dating based on plants, microfossils, seeds, leaf, and charcoal. On the left figures is C14 days, calibrate years before peak presence, and on the right figure shows the 8 models of using Bacon's Bayesian statistic-based routine that model accumulation rate by dividing a sequence into many thin segments and estimate linear accumulation rate for each segment based on calibrate C14 days. Also, based on the 8 models, you can see that there are hiatus in both sequences in CPTA and also in CP4. These hiatus are indicated by 8 models and also indicated in the sediment sequence as well. For the paleo environmental interpretations, I made a cross-section in south-southwest, north-northeast direction to illustrate the status of Lake Kumhawati during different time periods. The top of the figure shows the location of polling site of this study. The stratigraphy and proxy data indicate that Kumhawati was an open water lake and high runoff between 10,000 and 7,000 cal year BP during the early Holocene. When compared to other Paleo records from the Asian monsoon regions, this corresponds to the early Holocene intervals of a stronger summer monsoon by about 7,000 Kelvin BP. The southern part of the lake had transformed into a peatland, while a water body still occupied the deeper basin until about 6,500 Kelvin BP. This signifies a shift to lower effective moisture, which is likely a result of a weaker summer monsoon. The driest interval in Kumpavapi history possible occur after this period. However, the presence of the hiatus along hiatus means that the sequence of Lake Kumpavapi does not provide paleo environmental information for the time interval between 6,500 year to 2,000 Kelvin BP. The second lake phase was established again between 2,000 to 1,400 Kelvin BP, which point to higher effective moisture availability. The lake was of a smaller size than the lake that existed before, since peat goats still continue to be present in the northern part. The chronology of Kumpavapi cannot give detailed information about paleo environment during the last 2,000 years. Therefore, we focus on the study of Lake Apo for this time period, for the last 2,000 years. We did analysis multi-proxy of Lake Apo, similar to Lake Kumpavapi that I showed the result before. But in these figures, I selected only delta C13 isotope as a representative of moisture availability related to monsoon, and the charcoal as an indicator of fire or human activity around the area. This table on the left-hand side shows the settlements, archaeological periods, and human activity in Northeast Thailand. The cultural sequence from this side can be compared during Bronze Age and Iron Age. People start cultivate during Bronze Age in these regions. Water management are important feature of the Iron Age in Northeast Thailand. We focus on the detailed study of Lake Apo and Iron Age in Northeast Thailand. People in the past have settled to where you can get more water. For example, around freshwater lake or river, they adapt themselves to environment and leaves some evidence in the landscape and geomorphology. For Northeast Thailand, you can find mount, mock sites, reservoir, and canals. It's important. It's an example of how ancient society responds to monsoon variability. In archaeology, it still discussed why the mock sites or mounds during Iron Age were constructed. Was it a response to drought or to a wet period and flooding? A mound is a small hill. Moat means the combination of mound with constructed channel around mound. Some authors interpret the construction as a reflecting severe drought, while others argue for several other possible environments and societal factors, for example, defense or hierarchy. We go back to this side. When the Iron Age chronology for Northeast Thailand is compared to Bako record here, we see that higher effective moisture or wetter period likely characterize the early Iron Age phase during Iron Age 1 and Iron Age 2, and that it was followed by dire conditions during late Iron Age. According to archaeological data, The mounds and mob sites were built during the early Iron Age, when the data set for Paco suggests a stronger summer monsoon, but more intense channel networks were constructed during the late Iron Age, when the summer monsoon seemed to have weakened. There seemed to be a link between the climate and human settlements. Humans depend very much on water with a secure food and water supply. Settlements, growth, economics, and territorial expansion became possible. So from my study, I have the brief conclusion, the message to take home from the Lake Kumpawati and Lake Pakhon. Sediment sequence and proxy from Northeast Thailand can provide a detailed picture of lake wetland response to hydrological change. The Holocene record from Northeast Thailand adds importance paleo-environmental and paleoclimatic information for us for a region in Southeast Asia. Stronger summer monsoon are indicated in the interval early Holocene 10,000 to 7,000 kVP, and weaker summer monsoon during the mid Holocene and also during the late Iron Age phase, about 650 to 450 kVP. Changes in summer monsoon pattern had an impact on ancient society in Northeast Thailand. For the next chapter is about speleothems. What is speleothems? Speleothems, commonly known as a cave formations, which are secondary mineral deposits formed in cave. For example, maybe you know stalagmites, stalactite, column, and foldstone. But scientists mostly use a stalagmite for paleoclimate studies. Above the cave, rainwater passed through soil and carbon dioxide gas from plant respirations, creating carbonic acid. This weak acid passed through joints and cracks in limestone bedrock above the cave. The mineral calcite is dissolved from the limestone rock. When dissolved water is exposed to the air in the cave, it releases carbon dioxide gas. Calcite is precipitated on the cave wall ceiling, and when it's precipitated on the floor, it's called stalagmite. For the advantage of studying stalagmite, from the sample here, you can cut the stalagmite into half. You can do absolute precise date using the uranium-thorium dating technique, and you can also analyze various climate proxy, for example, delta C13 isotope, delta O18 isotope, and test element. Also, this figure shows overview of location of investigated speleothems that were used for paleoclimate study around the world. The database are collected by the effort of the Pages Science Working Group. In the current map, you can see that the red square there. There is no speleothem study in mainland Southeast Asia. Until 2019 and 2020, there are 3 publications from Thailand and Laos in the bottom. And I will tell you about the story about the speleothem study in Thailand. Cave survey in Western Thailand is quite challenging in terms of accessibility in some part of the area, and also you meet a lot of new friends. And also it's also challenging in terms of the result, and I'm going to talk about my data now. After survey, we find a good stalagmite sample for paleoclimate study. We cut it into half in the figure that I show on the slide here. And for the uranium-thorium dating, chemical separation of stalagmite sample need to be done in the chemistry keen laboratory. I don't have it. We don't have it in Thailand. That's why I have to do all analysis at the Earth Observatory, Singapore. This is supported by my colleague, Professor Chen Fong Wang. And there are several steps of digestion until you get only the uranium and thorium to be measured by multi-collector ICP-MS. Then you get the result of the dating. Here you can see on the left-hand side at A figure in A is a half section of stalagmite with location of dating in the white color. And the result in B is a star 8 models and C bacon 8 models. Maybe you are shocked when you see the big error. Like if you are working with chronology, the blue shape shows the date with 2 standard deviations. The green line indicates the likely 8 models, and the red line shows 95% confidence range of the 8 models. Our sample have quite large error because of the nature of the bedrock. We have a high thorium contents due to argillaceous limestone as a bedrock or impure limestone, which is the nature of the bedrock in Western Thailand and parts of the Myanmar. That's why it's quite difficult to find a good sample for paleo climate study. But it is the best we can do from what we have. This sample dates back to about 2,000 years. But based on the figure, you can see here that after 15 centimeter, you see the steady deposit of the stalagmite, and the chronology is let's say that better from that part. Therefore, we do a more analysis from the top 15 centimeter of the sample. And what we did the test elements analysis in here is a profile in the red line here, and we got the concentrations of test elements: iron, manganese, zinc, and copper. What we observe that the dark color layer in stalagmite, you can see it in the figure, correspond or show higher trace element compositions almost in every darker layer here. And then we also did some other analysis. The upper one is the scale value, which is related to the color of stalagmite. The second panel is iron concentrations, and the third panel is delta C13 isotopes. We observe that when you have a darker color in stalagmite, you have higher iron contents and you have a lower delta C13 isotope. At the same time, you have also some part correspond to lower delta O18 values. Stronger rainfall conditions enhance vegetation density and soil microbiome productivity, as well as lower amount of pre-calcite precipitation, which all can lead to lower stalagmite delta C13 values. Both the high trace elements and lower delta C13 values observed in our stalagmite layer suggest strong rainfall at the Cape site. And for the O18 isotope, you may ask, how can we interpret deep data in terms of climate? We did monitoring for 2 years. The data show from the we did the rainfall monitoring. The data show a broadly negative correlation between monthly means delta O18 isotope of rainfall and rainfall amount. It's isotope values. And also the model has been show the rainfall moisture come in our location of the study site. Most of 85% of the moisture come from the Indian Oceans, and only about 15% come from the Western Pacific during October to December. Also, that's why we interpret the delta O18 isotope values lower delta O18 isotope values mean that stronger summer monsoon. Okay, then this is the changes that we observe in our study site, which maybe is a local signal. Then, to better understanding of spatial pattern of hydroclimate change, if the local signal that we capture is can be compared to the regional patterns or to the larger area. We compare our stalagmite multi-oxy record with other paleoclimatic records in the Asian monsoon region. And also we compile the data of published data. The wetter and drier interval or more or less rainfall presented it follow the implication of the original publications. You can see the bar scale at the bottom of the figure that we compare with different proxies. What we observe is for the last millennium, we observe the opposite hydroclimate patterns between records from India and mainland Southeast Asia in the north, and opposite pattern with the Indo-Pacific equatorial region in the south. This may explain by the location relative to the migration of the IPCC. However, the north-south migration of IPCC cannot explain the similar pattern between records from South China Sea and from Sulawesi and forests in Indonesia. Also, therefore, we suggest that the Indian summer monsoon was dominant control on precipitation variability in the western part of mainland Southeast Asia in Myanmar and Thailand. But Pacific water circulation may have been the overlap control on precipitation on the site located close to East Asian summer monsoon and West North Pacific summer monsoon. For example, for the record in Vietnam on the site. Also, for the coast overview, I'm not sure if the time is that. Yeah, the coast view of the skeleton record. And also, we also not only compare with other paleoclimatic records, we compare the time interval of civilizations and history in Southeast Asia as well. I have the brief conclusion for speleothem study that a course view on the civilization and cultural history in Southeast Asia in the context of Indian summer monsoon variability infer from stalagmite record show an evidence coherence between hydroclimatic rhythm and the rise and fall in civilization or kingdom in the regions, whereas many other factors would certainly have affected the cultural history. The hydroclimate linkage we discover here suggests that climate change might have played an important role. Thank you for your attention. Not so sure about the time.
Thank you very much. Thank you very much, Moon. That was really interesting, full of interesting facts. I wonder if there are any questions. I can't see any on the Zoom chat. I'm just gonna check the none on the YouTube. So, Linda, do you have any questions? If not, I have some of my own, but I want
there is one.
Yes.
Yeah, there is one question in the chat box now. What is the sedimentation rate and the extent of bioturbation in the lake?
What is the accumulation rate of the sediment?
Yes,
Yeah,
you can read it in the chat box if you
I
see
think
that.
that you mean this one, the chronology, like, yeah, we have a big hiatus. The accumulation rate at the early Holocene is quite high, and then it suddenly disappear. Like the the model indicate hiatus here because it's very low accumulation rate. And then they have some higher accumulation rate again in the late Holocene. And this hiatus is indicated not only by the dating, but also we do see the change in stratigraphy. We do see the compact in peat layer and also the fine grain sediment. We think that it's likely the lake die out, it turned to be peatland. And when the lake die out, it's turned to be peatland, it also can lead to biodiversity and loss of some part of the peat.
Thank you, Mu. What about the extent of bioturbation in the lake? What do you think? Is there a lot of disturbance in the lake sediments? This is the other part of the question, the same question from Dr.
I think that nowadays you mean nowadays or in the past.
I'm not sure what the question refers to, but I guess that it just means, do you see mixing of the sediment or, reverse in the H model or any indication of bioturbation in the samples? I don't know.
For the early
Holocene, yeah.
From the early Holocene period, I think that the stratigraphy and the dating show quite no reversals in the dating and sediments, but you can see both. But during the time of hiatus, when the lake die out and it's some part of sediment and peat exposed to the air, it's oxidized, and then I think that we lose some part of the material, and we do see some disturbance and also mixing with the date.
I wonder if that's answered the question, Dr. Subramhushu. Sorry, I can't say it. Are you happy with that answer, or do you still have a question?
In fact, that answer is not very clear to me.
Could you clarify the question, please? Yes.
Yeah, first thing in this, to understand the present day lake sedimentation, one must attempt this late 210 dating, because with a half-life of 22.4 years, at least in the last 100 years, what changes have taken place, you can measure. And the second thing is that, then the second thing is, you talk about this carbon-14 isotopes, and it is severely affected by bioturbation. If you have read this paper by Turekian and others, long ago they have done it. So that's why I put this question, whether you have taken all these aspects into consideration.
Yeah, thank you for your comment and suggestion. That's of course that late 210 is a good dating method for the younger part of the sequence, in the top part of the sequence. And this study, I did it during my PhD. And I think that we think about to go back to the lake again and collect some surface sediments and do the different dating method, like late 210 is a good suggestion. But you know, the problem in Thailand, I don't know if the same in India, that people modify the lake. People already dig lot of sediments from the lake. Nowadays, the lake is not natural lake anymore. Because when you have a lot of vegetations and you want clear water, then people surrounding the lake or governmental officer or whatever dig the sediment out. Yeah. And nowadays, Kupawapi is one of the high tourist spots. It's very beautiful lake. You can see a lot lily water and lotus, pink lotus there. And surrounding the area of the lake have been modified by humans. But I think that it's good to come back to study the taste of human activity there, maybe with some other things.
Thank you.
Thank you.
Yes, please, Angela, you take. There was another question in chat box.
Yeah, Mu, thank you for that. So there is another question from Dr. Saju Menacheri. Do you have any idea about, this is about the speleotherm, so we are moving on to the second part.
Yeah.
Any idea about the temperature at the time of the stalagmite formation from your isotope work?
Yes, in our, I have to say that I'm quite new in speleotherm research as well, because I start to work on speleotherm after my PhD, and I get a lot of help from colleagues in Singapore. And in our publication that we publish, we try to calculate the temperature based on the equations of previous publications, and we calculate the temperature about 23 degrees Celsius of the is a temperature where it's possible when the speleotherm visited. But we don't see the what to say that in tropical region we don't see the big change in temperature during the year. It's not like in Europe that you have winter and summer with a high difference temperatures. Have I answered your question?
Dr. Saju Menacheri, are you happy with the answer? Would you like to ask anything else? That's fine. Thank you, Mu. I'm just checking the YouTube, but there is no comments there. So, Linto, do you have any questions? I have some questions myself, but I'll wait until you go ahead if you have any.
Mo, I just have one question. Thank you for the wonderful presentation. You have shown both sides, the lake core and also the stalactites. I was perhaps I wasn't clear about what you've mentioned about the error in the stalactite, that uranium thorium age. So why could you please tell me why is it so high that you have such a big error?
Yes, because of before you do the dating with the uranium thorium dating, the composition of stalagmite is calcium carbonate. But you want to digest the uranium and thorium out of calcium carbonate. It means it's like a test element inside that. But in terms of the impure limestone, you have clay together with limestone that contain some thorium. And for the people who do the research of stalagmite, they prefer to have a very clean sample and you have a high uranium content to compare to thorium content. But in our case, we have quite high thorium content. That leads to the error in our dating.
This clay you mentioned, is it that in situ formed when the water was dripping down, that clay?
Yes, I think that it can be both by the bedrock themselves. When the limestone, impure limestone that contain both calcium carbonate and clay, then it dissolves and come together with the dissolved water. And it's, in some study, they also said that it can come with the soil above the field as well. If you have really high weathering above your bedrock.
Thank you.
Thank you. I'm going to ask you something about the speleoterms. It's not my topic. So, but when you showed where your site is located in the map
Yes.
for the speleotherm work, it seems to be right on the edge of two water sources, right? When you were saying that the model shows that 85% of the water is coming from the Indian Ocean, and I don't know if you have that figure there. You know, it's in between the blue dots. I assume the blue dots are those coming from the Indian part, and red dots are the others. And it seems to be right on the edge of the two. I just wondered if you had assumed that that was the case throughout the history of the site, or if that boundary may have moved. I just, you know,
because
wondered if you had considered this.
if I understand the question right, actually mainland Southeast Asia is located between two Asian monsoon subsystems, the Indian summer monsoon and East Asian monsoon. Based on Wang 2005, he divided these two monsoons is about 105 degrees. This is about here in the eastern side of mainland Southeast Asia. And you see that we are in between these two monsoon systems, and we are located in the Pacific, western side of Pacific, and also we have also the ITCC that move across the our region. That's why the climate in this region is quite dynamic.
So would it make any difference to the isotope signal if your site you receive the you know, was in one monsoon or the other? It would make a difference to the isotopic signal or not?
Sure, because I think that the interpretation of the delta O18 isotope, if it's that representative of the amount of precipitation or the source of
yes, exactly. Yes.
precipitation, if you have two different sources mixing, that is complicated. But in our case, we think that also major the major moisture are from Indian Ocean.
Okay.
What we see in our signal.
Okay. So you think that was probably the case throughout the history of the 2,000 years that you've studied, or 1,000, I can't remember, 1,500 years that you've studied, that was the case. Okay.
Yeah.
Okay. I'm sort of keeping an eye on that, and also the watch, because I think that our next talk starts in 2 minutes. So probably, unless there are any more questions, I'm gonna clap myself. Thank you very much for your presentation. Really interesting. Thank you so much for joining us today. And well, I'll see you soon. And I will pass over to Linto to present our next speaker.
Okay, thank you so much. Nice to meet you.
Thank you.
I think we should start with the next speaker, Dr. Runa. Dr. Runa Anthony is a scientist at National Center for Polar and Ocean Research. She has done her PhD in marine sciences from Goa University, India. And since 2011, she is a scientist at NCPOR, Goa. She has got numerous awards, including the prestigious award like SERB Women Excellence Award, Young Researchers Award from Ministry of Earth Science. I also see that she was a recipient of SCAR Fellowship from the Scientific Committee on Antarctic Research. With this short introduction, I invite Dr. Runa Anthony to do her presentation today. Thank you. Over to you, ma'am.
Thank you, Linto. I hope you're able to hear me clearly.
Yes.
Okay. So what I'm going to be talking about today is slightly different from what you have been hearing for the last past 2 days, because in that I will mostly be talking about processes that are taking place on the ice sheet in the present. But you will see that in some way at the end of the talk, all these processes have implications for the interpretation of ice core records, as traces of past climate. So just give me a second while I load my presentation. Yeah. Is that visible to all?
Yes.
Yeah, okay.
Yeah.
I'm going to be talking about biogeochemical processes on the glacier and ice sheet surfaces. Now, when we talk about glaciers and ice sheets, about 10% of the world's entire landmass is covered by them. And you all know that it stores about 75% of all the fresh water on Earth. Now, the cryosphere plays a very vital role also in the Earth system processes because it is important in mediating global climate owing to its high albedo and cold water, which basically drives ocean circulation. And they also control global sea levels and ocean currents and storm patterns across the world. And this cryosphere, the hydrosphere, the atmosphere, and together with all the biological communities within it are a very closely interactive component of a very complex climate system, which has both global as well as regional influences. Now, once these ice sheets were thought to be really too harsh and inhospitable to support any living thing, but the ice sheets are now known to be this gigantic reservoir of microbial life. You have like 1,000,000,000 billion trillion cells, microbes in the top 2 meters of the Earth's ice. It's almost like a frozen rainforest. And it's not that empty white wasteland that people actually imagined it to be. Because if you look at the microscopic scale, there is an abundance of life. And the microbes living here have to overcome a lot of stresses like low temperature, desiccation, fluctuating light levels from the continuous light in polar summer to continuous dark in the winter. And they also have to deal with elevated levels of UV radiation and limited availability of nutrients. But these organisms have learned to overcome these stresses, and they are very active at these very low temperatures. And although hundreds and thousands of miles away from where we live, these organisms actually have a huge impact and significance for everyone on Earth, as I will show you in the coming slides. How they play a key role in the melting of glaciers and ice sheets, how they affect carbon dioxide concentrations in the atmosphere, and also cycle key elements and nutrients. And not just that, these organisms could also teach us about the potential for life elsewhere on our solar system. And when you look at the ice sheet from high above, it looks quite featureless, but actually it's often riddled with thousands of holes like this. And these holes are called Cryptonite Poles, and they actually form a main habitat on the ice surface. They look very simple, but they're home to a very rich ecosystem of organisms and are actually one of the most biodiverse microbial habitats on the Earth's ice. And these holes can range from a few centimeters to meters in diameter and depth, and they can dot an entire landscape. And these holes form when you have dust and debris on the ice surface that basically becomes entangled by long thread-like cyanobacteria, which are basically photosynthetic bacteria. And these cyanobacteria, as they grow, they start producing polymers, which are very sticky. And these act as biological glues, which will bind all the bundles of material together into very stable granules. And this bundling and binding of material creates a sort of microhabitat for other organisms, especially those that can feed on the molecules that are produced by these photosynthesizing bacteria. Now, as the granules grow and the microbes start reproducing and producing organic matter, especially humic organics, it makes these biological material especially dark. And because they are dark, what they do is, as you can see on the anything that is dark will absorb more heat. So they start melting the ice underneath, and it forms these sort of cylindrical melt pools. And it creates sort of a very stable habitat where you have liquid water available, and it's sort of a stable environment for microorganisms, have a protection from the weather and intense sunlight. And we can say that these cyanobacteria sculpt the ice surface and engineer a very comfortable habitat where diverse microbial life can thrive in these extreme environments. Now, another habitat on the ice surface is also the small, thin liquid water films between the crystals of ice. So when snow transforms into ice granules, you have a very thin, liquid layer which forms on the surface of the snow grain. And this liquid layer is enriched with ions and nutrients. And there you have bacteria and ice algae growing. Now, ice algae are very dark because they produce carotenoid pigments as a sort of protective mechanism against UV radiation. And this then darkens the algae and therefore the ice. Now, color of the ice, so you can see here how the color of the ice is quite dark. And this color of the ice is really important because the color would determine how much solar energy is absorbed compared to how much is reflected away. And the amount that is absorbed will determine how much energy is available to actually melt the ice. And explorers have really long tried to draw the attention of the public to the importance of life in these inhospitable regions. Even as far back as the 1870s, there was an explorer called Adolf Nordenskjöld. He noted that the ice on his expeditions were often brown or gray or black. And when he checked some of these ice samples under the microscope, he discovered there were traces of bacteria and algae. And he interestingly noted down in a section of his diary that microscopic life was the greatest enemy of the mass of ice. And until now, his notes have really remained obscure in the history of polar exploration. But in the 21st century, as climate science has become increasingly urgent, his work has gained new significance. And scientists have confirmed the presence of microbial life, not just on the surface of the Greenland ice sheet, but also elsewhere, and are now attempting to actually quantify the ice darkening effect. So this is a photograph of an area of the ice sheet in Greenland, and you will see how it is crevassed and pockmarked with these deposits of cryoconite. And you can see quite clearly here that these cryogenites actually turn the snow dark. And dark snow, like I said before, absorbs more sunlight and it accelerates the melting process. And one of the emerging concepts in this field really is that of bioalbedo. That is about how biological forces influence the albedo of the Earth's glaciers and ice sheets. And albedo is simply the reflectance of solar energy back to the atmosphere. So if something has a very high albedo, it's reflecting most of the surface energy back, so the surface can remain cool. But on the other hand, if something has a low albedo, it's absorbing most of the solar energy and it's becoming warmer. And in the context of ice, it's really bad news to have low albedo because it means that solar energy is being pulled into the ice, making it melt faster. And as microbes hasten the melting of ice, this is a positive feedback because what happens is the more melt you get, the more microbes can feed off the nutrients that are released within the melt water, and then you get more microbial growth. So this is really a vicious circle of how microbial growth and melt are driving each other. So these biological processes are very closely driving positive feedbacks in the ice. And that is a scary thing because current projections of how glaciers and ice sheets respond to climate warming don't really include this biological albedo feedback. And the first sort of experimental evidence that microbial activity can influence or significantly decrease the glacier albedo was provided in 2016 by Musilova et al. So a simple experiment was conducted to investigate the darkening of cryptonite debris as a result of organic matter accumulation that is driven by microbial activity. So basically cryptonite debris from Greenland, 10% of natural cryptonite was mixed with 90% of cryptonite, which was burnt at very high temperature to remove all the organic matter. And this mixture was then exposed to simulated Greenlandic summer conditions in terms of temperature and lighting and nutrient availability. So this cryptonite mixture basically simulates the early stages of cryptonite hole development where the debris is mostly inorganic and then it can become colonized by local microbial communities. So in the figure, you can see the green is basically samples that were kept under dark conditions, and the yellow are samples that were kept under light conditions. And there were, of course, amendments of nutrients like nitrogen, phosphorus, and carbon, which were made at ice meltwater concentrations. And the graph A shows organic carbon accumulated over one simulated summer season, and B shows the organic carbon accumulated over 3 simulated summer seasons. So the light samples, the yellow, you can see accumulated significantly more organic carbon compared to the dark samples. And this is, of course, because photosynthesizing microbes start producing organic carbon when exposed to light. They fix the atmospheric carbon dioxide. And this accumulated organic carbon over a period of melt season actually darkened the debris, as you can see in figure C, and it reduced the reflectivity of the ice. Over 3 simulated summer seasons, the reflectivity was reduced from about 30% to 15%, generating a very strongly absorbing surface. These experiments were basically the first to quantify microbially induced melting of the Greenland ice sheet. And the results of this study were then used in a regional climate model to project future microbially mediated increases in ice melt. And figure A shows the present biologically induced increase in melt rate in millimeters per year. And B and C shows future biologically induced Greenland ice sheet melt rates in millimeters per year. The more of the blue and the red basically means we have more of the melting taking place as a result of biological activity. And first order estimates indicate that the maximum microbially mediated Greenland ice sheet melt potential is about 17.3 gigatons per year at present, and it can go up to about 85 gigatons per year by 2100. So the biological impact on albedo plays a very important role in modulating mass loss from glacial surfaces. And it's important that these are included in albedo models if we want to adequately capture how glaciers evolve in a changing climate. And also it's believed that the warming climate will likely extend the melt season, which would then result in more biological activity, thus contributing further to darkening of glaciers and ice sheets. Now, these microbes in the snow and ice surface also play another important role, that of the cycling of carbon and nutrients, especially nitrogen. And the inventory of nitrogen compounds in the polar ice sheet is approximately about 260 teragram nitrogen, and it's largely dominated by nitrate in the much larger Antarctic ice sheet. But active biological nitrogen cycling does occur on the snow surface, wherein compounds such as ammonium nitrate and nitrite that are chiefly supplied by snow precipitation and wind are cycled through different pathways into organic nitrogen compounds. And the different forms of nitrogen are then available for other organisms to use. And being a cyclic process, organic nitrogen is also ultimately converted back into the atmosphere as nitrogen. And the width of the arrows in this figure basically represents the number of microbial groups that were associated with each process. Similarly, active nitrogen cycling also occurs in the cryocnite granules, as has been inferred from genetic studies. And in cryocnite granules, atmospheric nitrogen is fixed to ammonium, which is then aerobically oxidized to nitrite and then subsequently to nitrate. And this is later then reduced to nitrite and further to dinitrogen. So ice sheets also matter for the global carbon cycle because cyanobacteria and algae that are present on the ice surface capture carbon dioxide from the atmosphere and they convert it into organic matter. And there are other bacteria which then break down this material together with organic matter from other sources. And they then generate carbon dioxide that is released back into the atmosphere. So the balance between these two processes would actually determine whether a glacier is a net sink or a source of carbon dioxide. And this figure shows change in carbon dioxide produced and consumed over one ablation season in different Greenland ice sheet supraglacial habitats. That is, you have cryocnite debris, then you have tea ice, which is basically ice which has a lot of dust and debris in it. Also clean ice and water within the cryocnite hole. And all four habitat types studied were found to be active and autotrophic systems, meaning that they produce significantly more organic carbon through primary production than is being consumed by respiration. And this basically shows that microbial communities drive DOC production and recycling on the glacier surface. And to date, 3 attempts have been made to model this carbon flux on the supraglacial environment. And these are the values that have been generated. Now, what we have tried to do, as well as a few others, is that try to characterize really what kinds of organic material are present on the ice surface. And we have shown that there are basically 3 principal types of organic substrate on the surface of glaciers and ice sheets. One is organic carbon that are freshly produced by resident primary producers, and they're released as cell exudate or dead cell residues. And these represent the most labile form, like for example, lipids and proteins and carbohydrates. And you also have organic carbon that is transported from far away. This includes a more recalcitrant compounds such as cellulose and lignin or black carbon compounds that are derived from soot particles and other anthropogenic carbon substances deposited on the ice sheet surface. These would include your lignins and tannins and the black carbon. And then you also have, and these organic, allochthonous organic carbon is also utilized by some of these supraglacial bacteria. And you also have organic carbon from local allochthonous sources like exposed soils or rocks, the nearby marine environment, etc. And this is also likely to be mineralized by a relatively high portion of the microbial communities. Thus, we see that organic carbon on the ice surface are quite diverse. And we know that some of these compounds are labile and they are acted upon by microbes. And we also know from earlier studies that some components of this DOM or dissolved organic matter are photoreactive. But to what extent these microbial and photochemical processes actually impact DOM cycling on the ice surface is not known. So we carried out some in situ experiments in Antarctica. So field experiments were combined with mass spectrometry to basically show for the first time at a compound specific level what is the bio and photo lability of these distinct molecular groups on the surface. So what we did is we took snow samples and then we incubated them under different conditions. We had one set of samples where the microbes were killed and the snow was exposed to ambient light conditions. Another set where we retained the resident microbial communities and then exposed them to light to see the impact of light and microbes on organic matter transformation. And we also had another set where we retained the microbial communities, but then sort of incubated them under dark conditions so that we eliminate light and see what microbes do to this organic matter. And what we see is that the mass spectra prior to and after the different treatments are strikingly different. Like the black circles here basically represent the different molecular groups or molecular compounds that are present at the snow surface. And you will see in the light only that very few of these original compounds are present, and you have several new ones represented by the red color circles. And the similar thing happens in the light only and the light plus microbe. So we see that unlike the initial pool, DOM pool, the new compounds that are formed as a result of these photochemical and microbial processing have very distinct molecular composition. And you have new kinds of compounds produced by each of these processes. And so these processes in combination or individually play an important role in determining the composition of the DOM in these environments. We also see shifts in the nitrogen and sulfur and phosphorus content of DOM, which is of course not shown in this image. And these processes have also been shown to be important in transforming some refractory compounds like dissolved black carbon and carboxylic rich alicyclic molecules from the snow surface. What we find is photochemical processing of DOM both enhance the bioavailability of previously unavailable organic matter, as well as they can transform bioavailable compounds into biopersistent forms. Similarly, we find that microbial processes can form both photolabile as well as photopersistent DOM. So these processes may be important mechanism of DOM turnover on the glaciers, and previously unconsidered source of biologically labile nitrogen, sulfur, and phosphorus compounds. And so this has considerable implications for carbon and nutrient cycling on the superglacial environment, as well as substrate availability for microbial growth and metabolism. And these findings from the field are consistent with the presence of abundant and diverse microbes in snow from this region that are known to play key roles in the cycling of carbon, nitrogen, sulfur, and phosphorus. And they have been shown in the lab to have the capacity to degrade diverse organic compounds like proteins, lipids, carbohydrates, lignins, and so forth. And the implication of these findings also extend to broader glaciological studies, because the interpretation of ice core organic matter signatures as tracers of past climate or past environmental conditions will require site-specific understanding of the post-depositional photobiochemical processing of DOM, together with other factors that drive variations in organic matter concentration, as well as the composition in snow and ice. And our studies and that of others show that about 22 to 90% of this DOC is bioavailable. And over the past decade, this Antarctic ice sheet has been disintegrating at an alarming rate. And it is estimated that the annual release of glacier organic carbon via melting and carving to downstream freshwater as well as marine environments is about 3 teragrams. And these losses are expected to accelerate to about 15 teragrams of glacial organic matter, glacial organic carbon by about 2050 due to climate change. And this is equivalent to about half of the annual flux of organic carbon from the Amazon River. So this glacial organic carbon, such large amounts entering aquatic systems, is likely to stimulate microbial metabolism and ultimately contribute to carbon dioxide outgassing to the atmosphere. So it's quite clear that glaciers represent a very substantial reservoir of organic carbon. And as a result, the loss of glacier mass worldwide, along with the corresponding release of carbon, will probably affect marine ecosystems, particularly those that are surrounding our major ice sheets. So we basically now understand the major biological processes that are occurring on the surface of glaciers and ice sheets on a conceptual basis. That is, we know that microbes aggregate the supraglacial debris and produce dark compounds that can decrease the albedo and thus promote ice melt. We also know that these microbes can sequester carbon dioxide from the atmosphere and break down this organic carbon from various sources. And this microbially modified organic carbon nutrients are then exported to downstream ecosystems, potentially affecting the biogeochemical processes downstream. But still there are many blind spots and missing links in our understanding of biological processes on the glacial surface. For example, the influence of microbes on surface albedo has not been really assessed in a quantitative way, nor has the export of supraglacial organic carbon and its consequences for downstream carbon cycling. Another gap is the absence of quantitative and quite often qualitative data on inputs of microbial cells and organic substrates and other nutrients to these supraglacial environments. And finally, most of these studies dedicated to these supraglacial processes come from very easily accessible sites, and data from remote glaciers, particularly in the southern hemisphere, are lacking. Therefore, we have very little knowledge of the sort of large-scale spatial variations and biological processes on glacial surfaces. And integrated studies which will take into account whole glacial systems, which would account for, say, the carbon, nutrient, and energy fluxes, will be necessary for a more quantitative understanding of the climate cryosphere interactions. And the study of life, I would say, and the Earth's ice is truly interdisciplinary. And it ultimately requires all of us to recognize, as Norden Skoll did, the really intricate bridges that join the very big and the very small. Yeah, so thank you. That's it of my presentation. And if you have any questions, you can ask me, or you can even shoot me an email. My email address is given here. Thank you.
Thank you, Runa. Angela, do you have any question, or do you want to take it now?
There is a couple of questions on the chat. So I think Suryanish Pandey has a raised hand, so perhaps he wants to ask a question.
There is already a question there from Karthik also. So maybe Runa, if you may read it out.
1 second, I'm just trying to...
Runa, do you mind reading it so that people
yeah, just a minute.
can also hear it?
Yeah, I'm just trying to, not able to see it.
Just close, stop sharing, then it will be gone.
Yeah, okay, 1 second, just a minute. Stop share. Okay. Where are the chats? Okay, 1 second. Has human contamination introduced foreign microbes in the ice sheets? If so, will they interfere with the biochemical processes of the native species? Well, interesting question. Yeah, it is quite possible that human contamination would have introduced foreign microbes in the ice sheets, but increasingly there are a lot of protocols in place to make sure that this doesn't happen, but invariably there could be some foreign microbes being introduced. But the question is whether these microbes will actually be able to survive in the they're suddenly exposed to these really harsh conditions of the polar environment, and whether they're equipped to deal with all the stresses that are there. And we really don't know how well they would adapt to, and let alone interfere with biochemical processes of the native species. I mean, we really don't know that much.
There was one more question. I see that it's if the polar ice caps melted, what would it be in the underwater now?
If the polar ice caps melted, what it would be underwater? I'm not sure I understand the question. If you could elaborate on the question, I'm not clear what the question actually means.
Okay, meantime, shall I ask you some questions? I've got some.
Yeah, sure.
I found it very interesting. Yeah, I think, yeah, you may ask now, please.
Hello.
Yeah.
Yes.
Yeah, I just wanted to know the contribution of black carbon and aerosols, and how do you distinguish between the microbes contribution and this contribution, because this directly contributes towards the absorption of the albedo, and how do you distinguish these two?
So basically what we are doing is using ultra high resolution mass spectrometry method, where we can identify at the molecular level the different kind of compounds that are present. And based on the the hydrogen carbon and the oxygen carbon ratios, we are able to classify them as say lipids or lignins or dissolved black carbon, and then we are able to differentiate between what could be coming from, and several other chemical metrics to decide whether what could be coming from microbial activity and what could be coming from other sources. But again, it's very hard to distinguish whether the compounds that are resulting from microbial activity are actually how much of it is produced in situ versus how much is actually being deposited from the atmosphere.
Okay. Thank you.
Yeah, I think it's my turn now. Yeah, so what I was thinking is like a few things. I was quite curious that you see that straight crevasses that you see on the surface of the ice sheet. How is it formed? That's first one. Probably, I don't know, you may just say that. And another thing, if this cryoclonite granules, they causes melting, how long since it is being happening there? Because if it's just there for a long time, by now there won't be any ice. No, I don't know if there is any direct, how long since it is acting on the. Then one more question is how microbes reach there. Do you have any, like the atmospheric wind bring it to there, or how they get colonized in different parts of the ice?
Yeah, so microbes basically seed the ice surface through various sources. Yeah, they can come from the atmosphere, from through long-range wind transport. They can also be seeded from the surrounding marine environment. Like when you have sea spray inputs into the ice sheet, you can also have a lot of microbes being transported that way. And then also from local sources like the rocks and stuff. And once they colonize the ice surface, many of them quickly adapt to the conditions there, and then they start creating their own sort of ecosystem and communities there. And as to the question of how Cryptonite holes, you asked me that.
Since how long it's been there active?
Yeah, so these holes, so actually these holes are quite dynamic. A lot of times these holes are also connected to each other hydrologically through, say, sort of subglacial or endglacial melt channels, and many times these holes can collapse. Okay, releasing their contents into these sort of small streams, and then which then enter into the nearby marine environment. So once they collapse, they release all of their contents, and these larger debris particles within the cryptonite holes during this transport can sort of break down into smaller debris, colonize new ice surfaces, and from there they can grow bigger and bigger and form new cryptonite holes. So this process is like an ongoing process, and you will not find the same cryptonite hole in the exact same location the next, in your next expedition. You will find that it's not there, or several new ones, hundreds of them have found.
And the increase in deposition of these granules, is it related to some climate change or some,
Mostly these granules, basically, they are these dust and debris. You will see that most of these cryptonite holes you will find very close to sort of, like exposed rocky areas, and generally in the ablation zone of the glaciers. And mostly they come from the surrounding bedrock.
Thank you. Thank you for the answers. I think we have some more questions has come. Angela, do you want to take it?
Yeah, with the, I can read it. With the current rate of melting polar ice caps, how do you think that the rate of change in total biogenic carbon as related to microbes is varying? Yeah, so the thing is with, like I explained before, with when you have increased melting of the ice surface, you basically have more liquid water available to the microbes. Along with this liquid water, you have nutrients being released or more easily accessible to the microbial communities, which then sort of enhance the microbial growth. And this in turn has influences on the carbon cycling or carbon transformation processes on the ice surface. So these are sort of aspects that we are now beginning to understand, and where we are trying to we don't really have quantitative estimates of how this melting really impacts. We only know at a qualitative level that they do. But yes, melting would enhance microbial activity and therefore enhance the carbon transformation in these environments.
My video dropped off, but I dropped off Zoom, so I don't know if you answered, but there was another question on the chat about naming of the holes in the ice as potholes. I don't know if you can see it.
Oh, yeah, 1 second.
Slightly higher.
Yes, 1 second. We can also name that holes potholes or not. I don't know. I mean, they are called cryoconite holes because basically they are formed when the cryoconite. So cryoconite is basically the debris. So when the cryoconite melts into the ice, and that's how these holes are formed. So they are called cryoconite holes. So that's how the name comes.
Runa, I have a question, if I may. Thank you for a very interesting talk. I was just wondering if you just said a couple of things actually that might mean that you don't have an answer, but I have a couple of questions to do with where these cryoconite holes and where these sort of biomats are found on the ice. So the first question was, because they need liquid water, obviously, to survive. Do you know if they would be found, for example, in deep Antarctica, where the temperatures are very low? Mm-hmm. Do you know if they are found there? That's my first question.
Yeah, microbes have been isolated even from the South Pole snow. It's just that liquid water makes it a more conducive environment to live in. And so microbes flourish a lot better and more than in the dry, surrounding snow.
Okay.
That's why they are more habitats. Yeah.
And my other question is related to one of your latter slides that, I know that you said that we don't know actually the sort of total extent on the ice, but I was just wondering, compared to other biomes, how much carbon do we know? Do we know how much carbon there is cycling in these systems compared to other areas of the Earth? So is it a big, I mean, I realize they're important anyway because of the darkening of the ice and the effect on albedo, but do we know if they're important in terms of, the amount of carbon cycling through the systems?
There have been a few estimates of the amount of carbon. If I can go back to my slide, I can probably show you. There have been like a couple of attempts to estimate the amount of carbon cycled on the ice surface, but the problem is this can vary a lot with the kind of environment. A sample, for example, the carbon cycling on the snow surface would be very different from that in cryoconite holes. In cryoconite holes, it would be several magnitudes of order higher. It would also be different in, say, surface lakes and melt pools, and also in the ice. And we don't have enough of data from all the different kind of environments that are there on the Antarctic surface to basically quantify what accurately what is the net amount of carbons that is cycled through these systems. And also we are only accessing or sampling certain areas which are easy to access, and so we miss out on a lot of the important regions. Yeah.
So do you think the way forward is to have some sort of remote sensing assessment, or is it hard with remote sensing only? You need always ground truthing and sort of samples.
Yeah, because with remote sensing, it's really difficult to see even Cryptonite holes, because they're really so tiny. So you do need a lot of ground truthing. But of course, recently people have also been using drones which are fitted with pyranometers and cameras that can actually document these Cryptonite holes over a much larger area than you could do by foot.
I guess you can access more remote areas, but still, the drone can only go so far, I guess.
Yeah.
So some remote areas, we
Yes.
still cannot access the drone. Yeah. Okay. Thank you.
Thank you.
I haven't seen questions on the YouTube chat so far. Linto.
I think is it the time to go to the next session, or do we have
no,
some more time?
We have a little bit more time if you want to ask more questions.
I'm done. Thank you. Very nice presentation from Dr. Runa. Thank you so much.
Thank you. Thank you.
If that's the case, should we break for 5 minutes? Angela,
yeah, we can break it. We can give people maybe a break and Dylan can get ready. Thank you so much, Runa. That was an amazing, really, really interesting, fascinating talk. Thank you.
Thank you so much.
Yeah.
Thank you.
We clap. Thank you.
Thank you.
So we then have a little break. The next meeting, I'll just check to make sure that I'm not incorrect in thinking that there is 10 minutes now till the next meeting. Yes, it's in 8 minutes. So we have an 8-minute break. People can get a drink or do whatever and then come back. Don't go away. Very interesting presentations coming still. Thank you.
See you soon.
Hi, Dylan. I see that you have joined. I wondered if you wanted to start sharing your screen, perhaps.
Hi, sorry, I just need to change some preferences apparently to allow Zoom to access my screen.
That's great.
Don't
Don't worry, you have still time. We are early. We are on the break, so you
Break
have time
now.
to
Yes.
I'm gonna just go and get a cup of tea in the meantime. So I'm gonna switch my screen off, but I'll be back on time to introduce you, Dylan.
Okay, hopefully I'll have sorted it out by then.
If not, Christchurch has a really good team that can help you, I'm sure.
Right.
Dylan, do you want to try sharing now? Screen sharing?
I'm waiting for my system. I've just started. I use a Mac, so I'm just starting my system preferences up, but they seem to be taking quite a long time. As typical.
Yeah, we'll be here. So if something has to be done from our side, we can do it.
Yeah, no, I think it's me. Yeah. I thought I would just be able to. I don't normally use Zoom. We usually use Microsoft Teams. So.
All right.
It just seems to be hanging there at the moment. So I'll let you know as soon as it's.
Fine, no problem. Yeah, it's all right.
How's that? How's that? Is that okay?
Yeah, it looks fine. It's good here.
If I, if I flip something up now for me to look at while the present, can you see that as well, or can you just see the PowerPoint?
Yeah, we can see the PowerPoint now.
Can you see that I've got some notes on top of that or not?
Yes, it's there. Your name, and we can see you also.
No, sorry, what I mean is, can you see that I've opened a file on top of the...
No, it's not there. It's not there. We just see the presentation.
Oh, great. Okay, that's fine.
Yeah, okay.
I didn't want to block off the presentation with...
Yeah. Do you want to change the slide and see if it's working fine?
Sure, yeah.
Yeah, it's good. Yeah, it's fine.
Okay, so I think that we can start now. It's time.
Yes.
So I'm very happy to present Dr. Dylan Young. He is a postdoctoral researcher at the Geography Department in the University of Leeds, and he's going to talk to us about using proxy paleo data in a process-based model to explore peatland development over decades to millennia. And I ask him to do this to show us how all of the proxy data that we've been listening about can be actually used in models to predict the future, basically how things are gonna hopefully we can say something and mitigate climate change. So this is the idea of inviting Dylan. So thank you very much, Dylan, for accepting to give a talk in this session.
So as Angela says, I'm Dylan. I work in the School of Geography at the University of Leeds, and I've been working on what we call a peatland development model now for quite some time. And so I was going to talk about how it can be used from a paleo perspective. Some of what I've got really focuses on the past, but also we can see how that might be used to think about what happens in the future. So I think it's important just to start off and say that we currently use data from peat cores to reconstruct peatlands histories, and those reconstructions look back in time, and the models themselves, they can help us simulate the effects of future climate change and land use on peatland development. And they can also help us look back and say, well, what might have happened at a specific time, and compare that to our present day reconstructions of what happened in the past, so that we might be able to get more confidence in what we're looking at from the future. And that's because peatlands, as you know, are dynamic archives, and that means that those archives themselves can change over time. So what we might reconstruct might be different to what actually happened at the time. So in terms of what I'm going to talk about today, I'm going to give you some background to the model, spending a reasonable amount of time on that, but hopefully that allows you to see how the model might be used in paleoecology. Then we've done some limited work really on paleo proxies, but I'm going to say what the potential is in the model and give you some examples of that, especially in terms of the past, and then how that might link to the future. And then I'm going to use an example of something that we have done more recently, which is reconstructing carbon accumulation rates, and how we compare those to the actual carbon accumulation rates that happened at the time. So the model we use is something called Digibog, and this is a schematic of it. And you can see that we've got this, if you look at above this grey dashed line, we've got external influences on the model, which is rainfall and temperature. And then we use that to input essentially an effective rainfall. But above the line could also be different land uses. And then below that dashed line, we have the peatland itself, and you can see how those external factors influence things like plant productivity and peat decomposition. But then within the model itself, you can see that there's feedback, so how peat decomposition affects the hydraulic properties of the peat, which in turn affects water table behaviour, and that in turn goes back, and there's this negative feedback on decomposition. So these things interact within the model itself, and it allows us to see how those interactions might have played out in the past, but importantly how things play out in the future, because whilst we might have records from the past, we just don't know what's going to happen in the future. So the model itself is made up of columns, and those columns themselves are made up of layers, and I've tried to illustrate that in this diagram here. At the start of a model run, we don't have anything on the landscape, there's no peat at all, and so it grows up using these cohorts of plant litter. And there are other models that can do this. There's the Holocene peat model, which I'm sure some of you will know about. And the main difference between the model we use, Digibog, and those other models is they are mainly 1D models, whereas we can simulate landscapes in 2 or 3D. So we can get to see how the spatial structure of a peatland might develop over time. And the growth of that column, or all the columns within a model, is dependent on how much litter is added and how the peat decays. And so that can mean that columns grow, or it can also mean that columns shrink depending on that balance. We add a new layer of plant litter every year, and then within a year we decompose that plant litter and the peat in all of the columns in the peatland. And both litter addition and decomposition, they're affected by the water table position in the model and in the air temperature itself. So looking at one of the columns just in a bit more detail, we can see over time, over these 4 different time steps, how peat builds up. You can see at time 1 we've got this new litter added, and then by time 4 it's the layer at the base of the column, and it's become much more decomposed. And the function that we use for decomposition, you can see there on the right, and the decay affects peat permeability. So and that permeability in turn affects lateral drainage from the peatland. So it can be changed. That function is just a function that we use in some raised bogs or in blanket peatlands, but it can be changed to suit different peatlands and different peat types. It doesn't mean that deeper layers are always more decomposed. They can be, but they could also be quite well preserved. So it just depends on how that spatial structure develops. And also then to the left of that, the slide in this main column, you can see how we might get this profile down the column as you would do in a peat core. And then also we keep a track of the mass in each layer, so we know what it was when it was added, and we know what it is over time, and that helps us understand exactly what happened at various specific points. And then just finally on the structure of the model itself, the columns of the model, they sort of talk to each other in a way, and that's what determines how water moves between them. And that movement depends on the permeability of the columns around it, and also on the water table gradient between them. And the key part about this slide is really that these features of the model allow spatial properties to develop vertically and horizontally. So we're not just talking about this particular, a 1D effect. And then there's just an example now of what this might look like. So this is an example that I've done, which is actually a hill slope, but I've removed the slope from the plot, so it's just easier to see what's happening. It was run over 5,000 years, and if you look at the plot on the left, You can see how the surface of the peatland has changed in 1,000-year increments. I mean, you can look at this much more frequently. You could say, well, I want to see what happens every year. In this simulation, we used the climate from the north of England, and it just allows you to see how that shape of the peatland has altered over time and along the hill slope itself. And then on the right-hand side, we've got this plot of what is the mass remaining in each layer, so the proportion of decay that's happened in a layer. And to the far right, you can see this scale bar, which shows peat that's newly added or well preserved is green going through to yellow, and then well decomposed peat is there right at the bottom in dark orange or brown. And again, you can see how those properties have developed throughout that hill slope. We have still got some quite well preserved peat at the bottom of the base of the peatland, and then generally more decomposed at the margins itself. And I've just highlighted that particular column, and so if we were taking this as our peat core, these would be our records of past of humification of the peat. So we can simulate much more complex topographies than this if necessary, and we can also reconfigure the model for different peatlands. For example, I'm working on one for the Congo Basin at the moment. So that's essentially the model. I hope that gives you a bit of idea on how it works and what we can see in it. Then in terms of understanding dynamics of the peatland from the past and then how that might map onto the future itself, we can think of the columns in this model as the cores, the virtual cores of the peatland. So just there's 3 examples there from different points of the peat surface, and they can or already hold similar information as we would get from the peatland archive, and information that's of interest to paleoecologists. So we could see what the effect of climate is as in the past or in the future, and we compare them to the long-term records or to the recent past. We've done actually limited work on some of these paleo proxies, but we could have we can incorporate more of these into the model itself. So, for example, we can build age depth models from these virtual cores. We can look at water table dynamics. We've got plant functional types in the model, which we could configure for different plant macrofossils and use those as comparisons. You've seen we've got peat humification records, which is our remaining mass, and we could map that onto something like von Post or similar. And then we can recreate carbon accumulation histories as well. And we can then use those if we then say, well, what happens if we run some of the climate change simulations? How does that change the behaviour of the peatland and how carbon might accumulate in it, for example? And just on the right-hand side, I've got a couple of plots there from a paper we did in 2015 about how signals within the peat might be changed by different inputs of the climate or by the autogenic actions of the peat itself. So you can just see there's a climate here that we've used, which we've got the temperature and the net rainfall. So that could be past climates or it could be future climates. And then we've got a reconstruction of the water table itself. And again, that could be compared to reconstructions. So then just moving on, I'm going to sort of wrap up with this example of carbon accumulation rates. And I think a lot of you will be familiar with using peat cores to reconstruct what's happened in the past. But one of the key questions that we're looking at now is, well, what's happening in the future or what's happening in the recent past? And it's important because we need to know the effect on carbon accumulation of climate change. But we also know it's quite a tricky question to answer because, as you can see at the far right of these plots, there's usually an increase in the apparent rate of carbon accumulation in cores taken from peatlands, and that's the reason it's tricky to answer is because the peat in that part of a peat core is only partially decayed. So what it means is that the actual rate of carbon accumulation and the reconstructed rate are different. And that's, I'm sure many of you know, that's because when we reconstruct the carbon history of a peatland, we're doing it from our present day position, whereas the actual exchanges of fluxes took place at the time. And so that's when models like this could be really useful, because we haven't got a time machine to go back and see what might have happened, but we can use models to say, well, how can we see what has happened in the past and compare that to what we think might happen again, or might happen differently in the future. So you can see these three geographic regions, that they all show these upticks in carbon accumulation rates, but knowing what we know about this partially decayed peat there, what we're trying to figure out is, have things actually changed in this time period or not? So then what we want to understand is, firstly, does our model reproduce these patterns? So I've got an example, a simple example of that here. And the idea of this is just really to say, well, do we get similar patterns to the find in a real peatland? So this was a model set up as a raised bog over 5,000 years with an annual rainfall of just over a metre, temperature 7.5 degrees around there. It's a UK seasonal climate, but then the climate was repeated year on year, which is why we get these nice smooth curves. So you can see we get this uptick in the apparent rate of carbon accumulation as we get close to the present day. And the three lines on the plot just represent the three locations above in the peatland. There's the model of the peatland there. So we have the yellow column representing the margin, the green column is the center of the peatland, and then we have this point between the two, and you can see how those apparent rates have changed over time. So then there's a more, I guess, realistic simulation here, which this actually uses a climate input that varies not only seasonally, but varies from year to year as well. But essentially it's a static climate, and we can see how the blue line represents the apparent rate of carbon accumulation. So that's exactly how it would be reconstructed from a peat core. The calculation is the same. And then the green line is the actual rate of carbon accumulation. And that's akin to having a flux tower on the peatland for the whole of its developmental history, or having chamber measurements throughout that whole period. So as you can see, although we've got this uptick of this in the apparent rate as we get nearer to the present day, the actual rate of carbon accumulation of net carbon hasn't changed at all in this static climate. And there's a couple of reasons for that difference. One is the way we calculate the apparent accumulation rate, and the other is because of the time differences. So one is the apparent rate is from our perspective now, and the actual rate is what happened at the time. So it just shows us how we might use models to look at these differences, and how it could give us a bit of confidence when we go forward. So from this we can say, well, if we just use this reconstruction, it's very difficult to tell what the actual rate of carbon accumulation was. It's important to note that the peak carbon balance itself can be negative, whereas the apparent rate cannot. It's just part of the way it's calculated. So it's just highlighting some of the challenges that people are addressing now, actually, for how we might look at these for the future. So that's the real challenge, is when we're saying how is climate affecting current accumulation of carbon in peatlands, or the future rate of carbon accumulation, how can we know? And we need to use either ecosystem models like the one I'm showing you now, or other methods which account for these differences in the way carbon accumulation is calculated. There's also some very interesting work by Steve Frolking and others, where you can see similar comparisons. Yeah, so that's it. I'm not sure if it was a bit quick, but anyway, I wanted to thank all these people here, including Angela, who've been involved in this work over some time now. Yeah, so thank you anyway. And any questions?
Thank you, Dylan. Thank you very much. That was a really interesting presentation, and I hope it sort of highlights. So there is a lot of people, Dylan, you might not know, that are not peatland scientists in the audience. So the audience is very varied, but I hope it highlights how sometimes observations, the observations that we have in the Paleo record, are not as easily interpreted as we might think, and that we may need, quite sophisticated ways of looking at them. And I think overall, actually, over all of the presentations, some of the speakers have sort of highlighted this is a really good example. I hope there's a good example even for people that are not working on peatlands. So thank you, Dylan. I'm just checking if there are any questions. Linto, are there any questions in the chat? I can't see any on the actual Zoom chat. No, none that I can see either. Linto, do you have any questions, or shall I ask something?
You may ask. I have a question that is like this apparent carbon accumulation that you've mentioned. Why was it shooting up at the recent history? I don't know whether you explained it. I didn't get it completely.
Okay, yeah. So when peat accumulates, it decays at different rates depending on whether it's exposed to oxygen or not. So when it gets saturated, it decays less. And so in recent times, you have more plant litter is added and starts to decay, but it's not the actual final amount that will remain there. So a lot of what you see in that recent uptick will not be there in the future. So you have to try and account for that somehow. So probably, I don't know, in excess of 80% of what's there will be lost. And so you might have an apparent carbon accumulation rate of several hundred grams of carbon per square meter per year, whereas the long-term rates could be down in the 20s or 30s.
Dylan, maybe you want to show the slide where this is shown so that people can see what you're referring to. Yeah. Here. So these are uptick at the end.
Yeah, and it's generally, that's what you pretty much find because peatlands are dynamic archives, so this is changing all the time, and as that new layer of litter gets buried and saturated, it's the way it decays changes.
Yeah, and Dylan, I don't know if you want to talk a little bit about the problem that this poses for us, that we are trying to see if climate change is changing the carbon accumulation. But at this point, in the recent history where there is warming, there is this problem with the apparent carbon accumulation rate increasing. And, this is the problem that Dylan has been trying to resolve in a way, with the model. I don't know if you want to elaborate on this a little.
It would be nice to hear from you on that.
Yeah, so we're just not sure. So you can look at it and say, well, this looks like the accumulation rate is increasing, but is it? And the only way that you can really know is by taking the fluxes of the whole peat column into account, and that's not what happens when you calculate this apparent rate. But the apparent rate is what's observable now. That's the problem. So unless you have a flux tower which measures you know, the inputs and outputs of the peatland, then we've only got these observations. So that's why we've got this difference, and it's difficult to disentangle. So we're then saying, okay, we need to use models of some sort. If it's not a model like this one, which is, you know, as I mentioned, you know, a model of the whole ecosystem itself, some way of accounting for this decomposition and how we might get back to what the exchanges of carbon were at a specific time. So it's quite a challenge, but there's some good progress being made by various people, including Angela.
Dylan, I have a question. In one of your, if there are any, no, I don't think that there are any other questions. Early on, when you showed the humification or the amount of mass remaining in the peat, when you showed a 2D visual of the peatland.
Yeah, this one?
No, the one that was red and this. So what's the more humified versus less humified? Is red less humified or more humified?
More humified, yeah.
Why is it less humified at the very bottom of the peatland?
Because it's when the peatland is developing, those are saturated all the time, and so it's how the peatland starts to grow before it can actually before you get a water table developing below the surface. And then when you start getting that, then you get more decomposition and these this darker, more decomposed peat. I think one of the key points to note on this is right at the margin. So on that orangey plot on the left-hand side there, you've got that very decomposed column at the margin, and that's at the base of a hill slope. So what's happening there is, as water exits the model, that peat decomposes more, and so it actually then starts to form a barrier of moving more water out the peatland, and it allows the water table to build up behind the peatland, which enables the peat to stay less decomposed and builds up peat. Yeah, so yeah, it's quite interesting, and sometimes you get the patterns forming in different places. Depends on the peatland configuration.
On the configuration. And I had another question about the other slide that you were just showing with the climate quite constant and then the water table dipping. And I wondered if you could explain this. Yeah,
okay, so that's actually what we've just been talking about. In the initial years of the growth of the peatland, the water table is right at the surface, and then you start getting enough peat there to have the water table starts dipping below the surface, and that's what that initial drop is. So essentially it's what's known as the transmissivity. So it's the thickness of flow. It's the amount of water that can flow out of the peatland, and at that point it starts to increase and the water table starts forming below the surface. Does that make sense?
Oops, sorry, I was muted. Yes, that makes sense. Thank you. I'm just wondering if let me just check if there are any other questions, Dylan. Nothing on the, I think because people are not necessarily peatland experts, there's probably not very many questions, but I hope this, you know, it kind of shows how past proxies, you know, sometimes need careful consideration, really. Linda, do you have any other question for Dylan?
No, Angela, thank you. It was a very nice presentation. Thank you, Dylan.
You're welcome.
Dylan, I just a last thing. On the water table, so how you would use reconstructions, so how you would use paleo proxies in the model. You would normally compare your model output to those proxies, or can you use those proxies to drive the model? So can you use, for example, water table reconstruction to, tell the model this is how the water table was over time? Or, would you do it the other way around?
Yeah, so the water table, we don't impose any water tables in the model because it works it out itself. But we could tune the performance of the behavior of the model using Paleo records, and that would then help us with, when we're going past what's known into the future. If we've tuned the model for the past, then we could be more confident in what it shows us for the future. And we could actually simulate what happens to the macrofossils within the peatland. We can sort of put them into the model, and then we could compare those outputs with what you get from the peat core as well to say how well do they match up.
Ah, that's interesting. That is interesting. That's very interesting. So you could have, for example, tested amoeba. We had a talk yesterday on tested amoeba.
Yeah.
Introduce tested amoeba in your course.
That's right. Yeah.
Cool. That's very cool. Okay, well, I think that's it. Oh, there is a question now. So is in the chat, Dylan, I don't know if you can see it. It says, interesting, any significance of atmospheric temperature and carbon accumulation? So do you see any effects, I guess, of temperature on carbon accumulation in the model?
Yes, I mean, yeah, so if we increase it, again, it's this balance between decay, which is also affected by temperature, and litter production, which is affected by temperature. So it's just the balance between those two, essentially. So, but yes, we can model that and we can see it happening. Yeah, so that's why it's useful to have climatic inputs for the future, and if temperature is increasing, we could then say, okay, what's likely to happen to carbon accumulation.
Thank you. And I think it's answered the questions because Dr. Menecheri has just said thank you there in the chat as well. Thank you. I was just wondering, so could you so imagine that we wanted to know if a core, that has an apparent increase in carbon accumulation now, and we know the climate for that core, and we know we have some prediction of how the climate will change. We can take it into the future, and then you can see if the present carbon accumulation that we see is increasing because it's apparent carbon accumulation, if that would, be higher or lower than the, for example, Holocene average.
Yeah, we can do that. Yeah, I mean, sorry, I was just going to say it's important, to remember that these are, this is a model. It's not the real world, but it's so. I think it's useful to have it to be able to do this because we haven't got a better way of doing it at the moment, or we can't travel backwards and we can't travel forwards, so we have to use models to do this. But I think we also have to recognize that they are models.
I have one question, if that was like a significant, I don't know how the rate of compaction over the time that affects the calculation of accumulation rate. You may have some compaction, right, over the time if you go down in the peatland. Yeah,
so we don't have sort of slumping of the peat itself. We just have a layer gets thinner because it decomposes more, but we don't have any compression of the peat itself. So that's how the layer becomes. It doesn't become compacted through slumping, shall we say?
Oh, is it because of, even if you have more accumulation on top, that the lower level don't get compacted? Is that what you said?
It will. The level below, it doesn't become it depends what you mean by compact, but it doesn't, the layers all through the profile continue to decay depending on whether they're exposed to oxygen or whether they're saturated. But the layer gets thinner because it's losing mass, not because it's collapsing on itself.
Okay.
Dylan, we have a new question. Paul Tansley is asking, well, he says, great talk. And in terms of recent uptick in carbon, you said this could be dealt with by using long-term flux records. Do you know if there are any attempts to look at this as our long-term records grow? As we have more, eddy covariance, longer periods of eddy covariance?
I mean, I think people are doing that, aren't they? I mean, there's 20+ year examples in Canada, and there are other long-term measurements. That is the problem. They're just only a few decades at best, and often much shorter than that. So yeah, I think that's the gap that the model can fill. But yeah, so there's quite a big, and of course not all peatlands have flux towers or chamber measurements. That's the other problem with it.
Yes, I guess the Mer Bleu HPM combination is probably the best, right, in Canada?
Yeah.
That I know of, yeah.
Yeah, me too, yeah.
Thank you. Paul, did that answer your question? Yes. Okay, so I don't know. Let me just check if there's anything on YouTube. Nothing there. Okay, so I think that's it, Dylan. Thank you so much for sharing this with us, and a really interesting talk, and hopefully a good example of how it can be used in modeling. Thank you.
Okay, we should stop sharing.
Yeah, you can stop. We have actually our last speaker is from Canada, so we have a little break now because it's very early for her, so she will probably not be ready until the actual time where the talk is scheduled. So we have a little break now of about 13 minutes. So we'll start at 12:30 UK time. I don't know what time it is in India. Linked, what is the time in India?
It will be at 6 o'clock in the evening.
Okay, so a little break now until 6 o'clock Indian time. Thank you. Nicole, I just wonder if you might like to try and share your screen to make sure everything is ready for the for 6 o'clock
Hello.
time. Good morning.
Sure.
Good morning for you. What is the time for you?
7:20. So not too bad. Thanks for picking the late slot.
It may be that you need to get permission. Ah, good. Perfect.
Is that
works? Yeah, that's
full
perfect.
screen? That's good.
Okay. Not full screen yet. No, perfect. So you have 10 minutes to get a cup of tea if you want. I see you have maybe
Coffee.
already your coffee. Still, we'll wait. I think we'll wait so that everybody can join if there's some people that are joining specific talks. So we'll wait until the right time.
That's fine. I'll go top myself up.
Okay.
I'll leave the screen share.
Yes, thank you. So, hello everybody. Nicole, are you ready? I don't see your camera, but I assume you're there. Yes.
Coming. Yeah.
So I think we'll start now since it's the time. I just introduced you, Nicole. So Dr. Sanderson is a postdoctoral researcher at the Department of Geography of the Université du Québec à Montréal in Canada. And she has been working on actually dating of the peatland record, especially focusing on the recent past. So her talk is going to be comparing 20th century carbon accumulation in Canadian peatlands using classical and new basin age depth models. So she's going to, I'm really looking forward to hearing all of the latest advancements in lead to 10 and recent dating of cores. Thank you, Michelle. Thank you, Nicole, sorry.
Thank you, Angela. And thank you to the organizers of this colloquium for inviting me to give this talk today. It's kind of early here, so I apologize if the light changes as the sun's coming up. So good afternoon and good evening, wherever you are. You'll have heard a few peatland talks already in the context of this session. So yesterday with Tom's talk and just now from Dylan. So thank you, Dylan, for a really interesting talk. We're also fighting with apparent and recent apparent carbon accumulation rate terminology. So a lot to think about from your talk. So I'm pleased to be able to speak to you today about something different but complementary. Some particular challenges with dating recent peat accumulation, namely, as Angela said, estimating carbon accumulation for the 20th century in Canadian peatlands using different modeling techniques. So classical modeling and a new model called Plum that I'll talk about. So let's get this going. For those who may just be joining us and this is your first peatland talk of the session, or for those of you who love hearing more about peatlands always, I'm just briefly going to introduce peatlands' role in the carbon cycle and the focus on recent accumulation. You'll be able to see some very pretty pictures of peatlands from northern Canada. They're very different to peatlands in all of your other parts of the world. And I'm going to focus on my interest, which is very recent changes, so centennial rather than, and subcentennial rather than millennial changes. I'll go over methods we use to date these sediments, so explain very briefly like what we do in the lab, how do we construct these chronologies, what are the limitations with the different kind of models we use, and the precision and accuracy of classical models and Bayesian, the new Bayesian package Plum. And finally, very briefly, I'm going to go into some interesting and kind of pressing, in my opinion, implications related to these questions. So yeah, a bit of a different format for this talk. So you'll have seen a lot about peatlands already. They're defined as organic deposits of more than 30 or 40 centimeters, but they can go to meters deep, depending on where you are in the world. The organic matter accumulation exceeds decomposition due to the waterlogged, cool, acidic conditions. And peatlands in general are seen as archives of environmental change. They record vegetation changes, climatic changes, eco-hydrological changes as they accumulate over time. And in northern peatlands, sphagnum moss that you can see in all these beautiful colors in these photos are the building blocks of northern peatlands. So they make up the main vegetation type in the peatlands that I'm studying. Globally, peatlands store a lot of carbon. You probably talked about this a lot already, most of which is found in the northern boreal and subarctic regions. So approximately 1/3 of global soil carbon is in northern peatlands, which is equivalent to nearly half the carbon in the atmosphere, to just almost 2 times the carbon stored in the boreal forest worldwide. And the figure on the right, adapted from Crowther et al. 2019, I think represents this very well. Shows above ground and below ground carbon stored in above ground biomass in soil up to about 2 meters depth, according to latitude. So about 1/3 of that soil carbon is contained in peatlands. And northern peatlands are also known to have played an important role in carbon uptake from the atmosphere during the Holocene. So this figure is from the IPCC AR5, it's been around for a while. And international organizations like the IPCC, with their special report on climate change and land last year, are recognizing more and more publicly the potential of peatlands, their role in mitigating climate change. Oops. So how do peatlands actually absorb carbon? So they take up carbon through photosynthesis, they emit carbon dioxide and methane during decomposition. And as plant matter is accumulated faster than it decomposes in cool, moist conditions, this means that peat accumulates. So peatlands are a net persistent carbon sink. As we say, again, I'm going to hammer this home really. Peatlands are a globally important carbon store and a net sink, and have been a net sink throughout the Holocene. So the pressing question is, how will warming affect this peatland carbon balance? On the one hand, some studies are showing that peatlands, there will be increased decomposition as warming enhances microbial decomposition, whereas more recent studies, including one by Angela and a massive data synthesis by Angela, show that peatlands have been accumulating more carbon over the last millennium. And that factors influencing productivity, so photosynthetic active radiation, growing season length, mean summer temperature, and productivity factors are dominant. So here's where we jump into this dating topic. This increased rate of accumulation, and especially the recent part, is what we're trying to measure. Give you a bit of context of where the peatlands I'm studying are located. This issue of increasing peat carbon and the influence of climate on peat carbon is particularly important in Canada. So our peatlands contain roughly a quarter of the global peat carbon. And not only is the country itself warming faster than the global average, but northern regions in Canada are warming twice as fast than the rest of the country. And there have been massive shifts since the 1980s. So temperatures are increasing. As you can see in this little figure on the right, permafrost is also thawing and warming, more so in the high Arctic than in the subarctic. I don't know if you can see those colors from where you are. So again, hammering this home, carbon storage can be altered by really rapid processes such as climate change, which includes permafrost thaw. Also natural processes, drainage and flooding. In Canada, we have a lot of beaver dams. So peatland carbon accumulation on the scale of a few years can really be altered by beaver dams. One of our sites this summer, halfway through the summer, just beavers constructed a dam and flooded the whole site and changed all our measurements. But also anthropogenic land use change. So agriculture, restoration projects can also alter carbon fluxes and carbon storage within a peatland. So quantifying these rapid changes during the last century is really important to predict the future carbon balance and to eventually maybe mitigate greenhouse gas emissions. So to do this, we need to know how much carbon is there and how fast it is accumulating, or how much time has it been accumulating for. And the former, how much carbon, is a pretty straightforward question to measure, but the latter, how long and how fast has it been accumulating, is a big difficult question, a tricky question. So the chronologies are key in this case. So here's the question, well, you want to date recent peat, what can you do? So we've talked again about the importance of dating recent and rapid processes and the need to have these high resolution changes dated in the recent part of the peat. So radiocarbon dating, C14, is the most common method, but it's potentially costly if you want to have a good resolution, depending on your core or your samples. You can get potentially large errors within the last couple hundred years. And then there are some other issues potentially with calibrating the radiocarbon dates. So post-1950s, there might be some post-bomb calibration issues. So unless you can have a long series of radiocarbon dates, it's possible that dating a lot of cores could get fairly costly fairly quickly. So an alternative or additional solution, complementary solution, is to add some radionuclides or radioactive nuclide dating, which includes lead-210. I'll go through some of the basics of lead-210 dating. So some of you have already seen these slides multiple times. So lead-210 down here is part of the uranium-238 decay chain. Oh, circled it. So it can be measured in the lab, either directly using gamma spectrometry. It's a non-destructive process. Or it can be measured indirectly using alpha spectrometry of its daughter product, polonium-210. Polonium-210, which is a alpha decaying radionucleide, which decays at a very high energy compared to the other types of decay. So it's much easier to detect in certain low activity samples. So again, some of the basics. It's deposited into the peatlands through precipitation, mainly from the atmosphere. This is called the unsupported fraction, or 210 ppx. And this supported fraction, which comes from the bedrock or possibly from runoff, needs to be accounted for to be able to use lead-210 dating. So in ombrotrophic peatlands, which are primarily rain-fed, this unsupported, I lost my mouse there. This supported fraction should be close to 0. So I think this should work. There's this animation by Martin Blau at Queen's University Belfast that illustrates the process of lead-210 accumulating really well, better than I could explain it. So there's this black line, which is increasing, which is showing accumulating sediment. And the kind of orangey red on the left-hand side, which doesn't really change very much, is the supported lead. So you can see that this is a constant influx and it's very, very low. The blue record is the flux of unsupported lead, so that excess lead, which is incoming into the peatland at a constant rate over time, and this is the blue, but it's decaying over time, and the length of these blue bars changes with the sedimentation rate. So in the end, you get this profile where you have a fairly constant and consistent supported or background lead-210, and then this blue record, which is your unsupported lead, which you use to date your profile. So skip ahead back to that. We can also use another radionuclide, which is really commonly used, cesium-137. Cesium-137 is an artificial radionuclide, which is produced during nuclear bomb tests. So there are peaks that can be detected around the world. Depending on where you are, they're more or less pronounced. So the 1950s until 1963, there's an important peak of cesium-137. And then depending on where you are in the world, a Chernobyl peak is also detectable. However, from many studies and also my own personal attempts at using cesium in peatlands show that it's pretty mobile in peat. And so you end up with these kind of lumpy plateaus instead of really straightforward peaks. So if cesium works, great for you. But in peatlands, I wouldn't rely on cesium only as a backup for dating in this case. So a key assumption of all of these dating methods is post-depositional immobility in the peatland. So once it's deposited, it stays there. But that's the same with most stratigraphic studies. So I'll talk about modeling now. Firstly, classical modeling. So the most popular and widely used form of classical lead-210 date modeling is called the constant rate of supply model, CRS. So it was put forth by Peter Appleby, who's been working with this model for a very long time, since the late 70s. It's remained fairly similar, although there have been some revisions to the uncertainty calculations over time and the ease or feasibility of adding additional dating information to the method. So the assumptions of the constant rate of supply model are really flexible, and they allow for changes in the sedimentation rate. So the main assumption is that the rate of supply of lead at the surface of the peat remains constant over time. And then there's a little figure to show, kind of conceptualize how you calculate it. But basically the ratio between the complete total radioactivity inventory, So everything between the surface and the equilibrium depth. So the entire lead-210 profile needs to be accounted for. So it's the ratio between that and the inventory at whichever depth you're at. So what do we actually do? In the lab, whether you're using alpha or gamma spectrometry, you start by measuring your lead-210 activity or concentration in the lab, and then apply the constant rate of supply model to get dates. This equation I didn't say in the slide before, but it's on the down here on the bottom. It's based on the radioactive decay equation, so it's fairly straightforward. So once you apply your model, you get your ages, and then using these ages, you input them into whatever age depth model, software package, or method that you use. In this case, in my example, it's Bacon, if you know it. If not, it doesn't. It's not important at this stage. So you get your age depth model combining lead-210 dates and radiocarbon dates. So there's a few limitations with this method. Firstly, and out of order, I'm going to start from the bottom because I just talked about modeling dates. The dates for lead-210 in your final model are modeled twice. So first using the CRS model, and then again using your whatever model that you're using to get your age depth. So that's a very large source of bias. Secondly, because this model is based on the radioactive decay equation, it's an exponential decay model, like all radioactive decay equations, or most. So it always produces a logarithmic shaped model. So you can see in the figure behind this kind of little, well, this is an extreme case, but this little like kind of hockey stick, to use a Canadian term, a hockey stick kick at the end of the model. So the CRS model usually has forces this little kick at the bottom of the model, which isn't realistic always in most cases. And finally, and importantly, the model relies on this choice of equilibrium depth. So the point where there is no more lead-210 detectable in your sample. And this choice can be pretty subjective. You can kind of see at the bottom here, I have 4 points. So this is a fairly good model, but not all of them are so clear. And this subjective decision is quite frightening, actually. This study just came out earlier this year. These are results from a global Iaea inter-lab comparison. So a set of 14 labs around the world were given the same data set. So they didn't analyze the samples themselves. They were given a set data set of lead-210 dates and asked to produce age depth models using any of the classical methods. So CRS or CF or any of the others, but most of the models produce CRS outputs. And what this shows, the red dot in the middle of the graph on the left, graph A, is the cesium control point. So what this shows is that user decisions really matter because not all the models used the cesium in this case. And then even when using the same data, the same model, they've produced this enormous range of different possible ages for the core. So this, I think, highlights the importance of reporting which model was used, which decisions were used while constructing the model, and then also producing raw data for future model decisions. So while we were putting together some literature review on the latest Plum updates, we did find quite a large portion out of around 200 studies. There was about 30 that didn't even report which model they used to construct lead-210 dates. So that's difficult then to take the data again and apply it again. So for all future studies, hopefully everybody reports everything in supplementary materials. So to get around or to address some of these concerns, in 2018, Marco Aquino Lopez, who at the time was at Queen's University Belfast working with Martin Blau, produced a package, an R package called Plum. it's a package Plum, which is now available as an R package, but it is in other formats if anyone is interested in using different formats. So it's a model or a package that's designed for non-statisticians, non-mathematicians, paleoecologists like me, who find it difficult to construct new Excel spreadsheet, like big database spreadsheets or Python codes every time we're doing some age depth modeling. So this is a very handy package which uses a Bayesian approach to the age depth modeling. So by Bayesian, it incorporates prior information and likelihood distributions. So Marco couldn't participate today, unfortunately, but if anyone has questions about the model itself, I would highly suggest you take a look at the paper. It's very detailed, or you can contact him directly. I'll share his information again at the end of the talk. So according to that paper and the work that we did comparing different simulations and trying it out on some cores from my PhD at the time showed that it's an accurate integrated model. So you can integrate lead-210 dates and different kinds of dating techniques at the same time. It also has more realistic uncertainties. It's a Bayesian analysis. So any additional information or priors increase the accuracy and precision of the model. And the model itself learns from additional input data. So Marco is at the moment working on a paper that should be submitted before the end of the year, which is quantifying exactly how much more accurate and precise, or how accurate and precise is this model by running hundreds of simulations with random configurations of data. He also showed in a paper last year that it does provide similar results as the CRS model with much less user requirements and with more realistic uncertainties. It's an aquatic marine system, and there were 4 cores or 5 cores. So that came out earlier this year. So I'm interested to see what the data look like for peatlands. And it's, as I said, easy to integrate additional dating results. So if you have cesium or radiocarbon, it's very, very easy to put all these together and have an output. And the parameters themselves, so you can see in the formula here, there's this, for example, this phi parameter. This is the influx of lead-210. So all of these parameters can be modified as prior information if you have additional information for your site. So what happens in the while using Plum? Well, we start with these same activity concentrations and the same core as before, same methods, but instead of double modeling, you just take all the raw data, throw it into Plum, and you get an output. So here, I didn't explain the figure before, but here in the kind of shaded blue are the lead-210 activity concentrations that Plum has remodeled. And then in this curve is the age depth model itself, the resulting age depth model. These long purple blobs are radiocarbon dates, quite uncertain in this case. But in the red dotted line is the most probable, most likely model. And then the gray shaded areas are the other possible models, but they're less likely. The darker the shading, the more likely the model. So you can see where there are lead-210 dates, it's quite a very likely model because you have a lot of information going into the first top half of this core, and then the bottom half with just 2 radiocarbon dates, there's fewer, there's less information. So as I said, Marco is currently working on testing the accuracy and precision of the Plum model. So to do so, we have to compare results against known age depth functions, because if we're just comparing CRS and Plum, we can see that they're the same or they're different, but which one is more accurate is the question that we're trying to answer at this point. So Marco is working with the simulations, as I said, and I'm working with cores with known dating markers to try and evaluate these questions. So we have these papers that are in prep, so I'm going to give you a little sneak peek of some of the preliminary data. So So firstly, simulated data sets. Some of you may have seen an earlier version of this earlier this year. So first in these simulations, Marco set up these scenarios which use or emulate common sampling strategies. So for example, from left to right, we have a full data set where every depth is measured. In the middle, skipped samples or missed samples. So a sampling strategy where you might just sample every other depth. And the final strategy, background not reached or incomplete background. So you have a high resolution at the top of the core, and then towards the bottom of the core, you start spacing out your samples, for example, or you maybe ran out of core or ran out of money. So you just don't have the bottom. So in each case, the green line here, the green curve is the true data set. So because they're simulations, we know what the true data set should be. And the red lines are the constant rate of supply CRS model, and the dotted blue lines are Plum, the Plum output. And each of the two models has the 95% confidence intervals on either side. So the points at the bottom show for the same models the normalized offset from the truth or true data. Both models do deviate from the truth in all cases. You can see in these first two cases, it's very clear there's this, I was talking about the little logarithmic kick at the bottom, the little hockey stick at the end of the graph. So it's quite clear in these two cases. You also have widening uncertainty, or this kind of trumpet, very narrow uncertainties at the start of the core, and then this widening trumpet at the bottom in the first two graphs. The normalized offsets do vary a little bit, but the scale is quite small in both of these cases. So any divergence shouldn't be too worried about. And at the bottom of the skipped samples, there is a pronounced divergence of the CRS model from the truth. So the ages are overestimated compared to the truth and compared to Plum. In the background not reached sample though, simulation scenario, this divergence of the CRS model from the truth and even from Plum is quite large. It's a factor of 8, whereas the others are a factor of 0.5 to 1. So if background isn't reached, that's quite a serious problem, as we see from this scenario. So it's best to have a full core, but it's better to skip samples than to leave out the bottom of the core, is the moral of the story. So if you can have a better resolution throughout the either spaced evenly throughout the core or maybe towards the bottom, then that's the ideal scenario. Well, the ideal scenario is to have all of the sampling points. So what do these scenarios look like in cores? So I'll give you a sneak peek at some of the real core results. Again, this is preliminary work. I should highlight that Marco is rerunning these kinds of samples at the moment with different configurations of data missing. So they'll be run about 500 times each, probably 100 to 500 times, depending on the scenario. So to put you back into the peatlands context, because I feel like it's been a while since we've talked about them, just for scale, we have vast and dense peatlands in Canada. So as I said, almost a quarter of the global soil carbon worldwide. It's almost half of what is sequestered in the forest worldwide. And over half of these in Canada are located, and the densest areas here, for example, around Hudson Bay, are all located in degrading permafrost zones. So part of my job at UQAM, Université du Québec à Montréal, is to, I've been compiling this massive database on recent accumulation rates in cores for Canada, in peatland cores for Canada. So where we have really detailed records for the recent periods, so the last 100 or so years, the 20th century. But for the purposes of this exercise, where I have the most deal are cores from the Continental Paleo Ecology Lab. at UCAM. So I'm just going to focus for the next couple slides on some results from Eastern Canada, or Quebec and Labrador. Oops, sorry. I have a surprise slide here. My apologies. I'll just delete that one. There we go. So this looks quite horrifying. When I first plotted it a few weeks ago, I was a bit shocked. Well, not surprise shocked, but a bit horrified, let's say. So I've plotted here on each of the axes the ages resulting from the same cores. So I have 18 cores in this case from Eastern Canada. And they're the full profiles. So for example, this is one core you can see here. This is another. You can kind of just see this typical shape. So we have on the x-axis the constant rate of supply model, y-axis the plum outputs. Again, just to clarify, all of these outputs are unadjusted. There's no parameterization happening. So I haven't introduced any bias. They're just straight models. And they do not include any of the existing data markers. Date markers, so any of the control markers that I mentioned. So no radiocarbon dates incorporated, no cesium. So you can see there is quite a large divergence of what is happening, what Plum is doing, what CRS is doing. But interestingly, if you look at the cases in blue, these numbers should be reversed. This gray is 12 cores and blue is 6 cores. The blue cores represent those where the background was not reached. And the gray cores represent either evenly spaced data or continuous data sets. I haven't separated those out because some of them do skip data, but only a centimeter at a time. And so that would be another interesting exercise. So it highlights again the importance of reaching background and accurately estimating the supported lead. Since 1900, my graph has disappeared, but since 1900 during the 20th century, you can see here that the error, if you're excluding these cores, the error here is less on average to around 1900, less than 10 years. So not too bad. And that, beyond 1900, the ages are likely overestimated for the older CRS outputs. So we think that this gap above the 1-to-1 line is due to that logarithmic kick occurring at different parts of, at different depths and different ages for all the different models, hence the widening of this gap. So we're kind of getting the similar results as in the simulations. But it would be interesting, the next steps of this are going to be to model it with the depth markers. So I thought it would also be an interesting exercise to see how this translates to carbon accumulation rates. So again, same axes, the constant rate of supply model, and then plum on the y-axis results. And these little outliers here, the 3 points there, and then 2 on the bottom, everything that's below the 1-to-1 line, are again those models that did not reach background. So this is again highlighting the importance of reporting your data, reporting what you did to the model, reporting which model was used, because in reporting these carbon accumulation rates calculated with the different models, you can get a difference of around 100 grams of carbon per meter square per year, which is quite large. So, so hence the point of my talk, which is challenges with chronology. So to wrap up and to summarize everything that I've been talking about with some data teasers, but keep an eye out for any results, which will be submitted before Christmas. So some important implications for what we need to think about in constructing chronologies is that Plum is a really user-friendly package if you're friendly with R. useful for inputting data, applying age depth models in a really consistent manner. That's the important thing. The CRS model, as we saw from the results of that Barsani et al. Review, is not applied in a consistent manner. So Plum and the constant rate of supply model both perform very well if applied properly, as I said. And the case study in Marco's marine environments was very interesting, and I'm looking forward to doing the same in peatlands. The importance of accurately estimating the supported and background cannot be overstated. And that's a massive limitation with the constant rate of supply model, is that that is necessary for the model, or it needs to be inferred. Whereas with Plum, you can add in some additional data from the same site or modify the priors, although it is also a key factor. And most robust chronologies are always, as always, used when combining multiple dating methods. So post-bomb radiocarbon dates, cesium if applicable, tephra, etc. And this is done really easily with Plum compared to other models. And so in our opinion, Plum should be a go-to for studies quantifying rapid carbon accumulation rate changes for the last century. And so again, hammering this home, accurately dating the last 200 years is really critical to addressing the response of peatland carbon to climate changes. For example, anthropogenic pressures or permafrost thaw, as you can see in these photos. It's quite drastic. Well, yeah, so I'm happy to take any questions, but I'd like to highlight a big thank you to Michelle Garneau for letting me use her continental paleoecology lab database to start some of these LED210 trials. And then to Team Plum, of course, Marco, Jose Andres, and Martin Blau, who are around the world, but who developed Plum and who are always very patient with getting questions about the modeling itself. And then to all the students who contributed data to the database, the paleoecology database. So thank you. It was a very preliminary result, so I'm very happy to get any feedback, questions, ideas for what to analyze next or how to model some of the results.
Thank you, Nicole. That was a fascinating talk. And thank
well,
you.
You caught me at a few months early. I think in a couple of months we could have done some of the final results.
Fascinating talk now, and you know, loads of interesting information for anybody working on the recent past, I think. There is actually some questions already in the chat, and you told me that you can't actually see it, so I will read them for you.
I think I can see it if I click.
Can you see it? If I could ask you to read them so that the people following us on YouTube can also hear the question.
Sure. So what role does permafrost play in the subsidence of the peatland resources? Will a decrease in permafrost cover lead to an increase in total organic carbon content? Oh, that's the big question, isn't it? Yeah, thank you for that question. It's a very interesting scientific, but also policy question at the moment for Canada. So with permafrost thaw, what is talked about in the news a lot is with permafrost thaw, you get an increase in wetness, of course, and water availability, less ice, more water, which leads to an increase in methane emissions. And so that's talked about in the news quite a lot, or has been over the last 5-10 years. The methane bomb, like the methane bomb will kill us all. It'll make, it'll increase carbon in the atmosphere and have negative feedbacks with, or positive feedbacks, a feedback with climate change. And it'll make it worse. But so what we're looking at as part of this Canada-wide project, and there are other projects in Europe as well that Angela is working with, that are looking at the effect of, with permafrost thaw and increased water availability, moisture availability, less frozen ground, come more plants and more productivity. So while you have these, let's see if I can get a photo here. For example, in this kind of subsidence area over here, I don't know if you can see my cursor, but there in the bottom photo, there's quite a wet patch. So all of this mound on the right used to be a big ice mound, and as it's subsiding, all of the trees are falling in, and all of the peat is also subsiding. So from this wet patch, you get an increase in methane emissions. But around the edges, what we're measuring with our project is the increase in photosynthesis. So the lateral expansion, the influx of mosses that are coming to colonize from the edges in this case. So the big question then is, will a decrease in permafrost cover lead to an increase in total organic carbon? I can't answer that right now. We don't really have anything at the moment where everyone has mushed together methane emissions and photosynthesis carbon sequestration. There are models that are working on doing that, but they, as Dylan was saying in his presentation, they are models, so they can include everything, but we're getting some very interesting results coming from those kind of studies. So that would hopefully be a next step, and our database could go and inform some of these models. Does that answer your question?
Yeah, if maybe Soham Banerjee can maybe tell Nicole if that's answered the question. In the meantime, there is another question.
Yeah, I'll come back to your question if that didn't answer it.
Oh yes, thank you. She said thank you, or he said thank
great.
you.
Okay, thank you.
And
yeah, go ahead.
Do you see the next question?
Yes. So is there any role for surface vegetation in the dating of peat cores? Yes, well, that's interesting. So one of the big uncertainties with lead-210 dating that was raised maybe 6-7 years ago, 8 years ago, almost a decade ago, was that the necessity to include kind of the surface little scraggly shrubby vegetation into the lead-210 inventories. Because as I said, it's very important to account for the total inventory of lead-210 when using the constant rate of supply model. And if you have little shrubs or little plants with leaves, they might be intercepting some of that lead-210 that's being deposited from the atmosphere. So until those plants die and then become incorporated into the peat, if you do want to measure the full inventory, you should be measuring some surface vegetation, but that's up for debate. As for trees, I was assisting with a master's student project last year who we thought that because her peatlands were forested bog, that the trees would probably intercept quite a lot of the lead-210. And that was the case to some extent, but the dating still worked very well with radiocarbon dates, so it didn't interfere, in her case anyway, with a good lead-210 date profile. If you have any charcoal or bits of I guess a tree trunk or something, you could also use those to do some other forms of dating.
Thank you. Maybe Chima AS can tell if they're happy with the answer.
Thank you.
Yes.
Oh, I see another question as well.
Yeah.
Oh, that's a good question. Could you please explain how to prepare the sample for dating? Are there selection criteria? So no. So yes and no. I mean, they're not as restrictive criteria, or not restrictive, but selective criteria as radiocarbon, for example. Radiocarbon, you need to be very careful about what you're dating. So if you're dating a bit of wood, that maybe isn't a great thing to date if you have other options, because you don't know, is it a root, is it a branch, a bit of twig, where does the wood come from, when was it deposited? With sphagnum moss, the way that it grows, it's ideal for dating. For lead-210, it's not really a selective process like radiocarbon, because you're trying to capture the you're trying to represent the deposition. So if you're using a constant rate of supply model or plum, which also involves an influx, you want to be sure that the influx is well represented. So usually we just take half a gram or plus or minus dried sample and homogenize the whole thing. So as long as there aren't any like big rocks or twigs or anything in it, we just homogenize everything.
Great. Thank you, Nicole. Linda, do you have a question? I see that you've unmuted yourself.
Yeah, okay. I had a question that first thing was like, how deep can you go here so that you can use this method in your pit box? How deep one can go?
Yeah, good question. It really depends on the peat bog. So, or even within a peat bog. So I'll give some examples. For my PhD, I dated some transects of microforms, which were within a couple meters of each other. So it was four or five different microforms, depending on the site. And in a hummock, for example, where there is this mound of sphagnum, you can go quite deep. One of my sites had lead-210 till about 60 centimeters down, whereas some very slow accumulating microforms further north that have lichen topped, so the productivity is really a lot slower. Maybe the first 10 centimeters would have lead-210 in it. So this is why we're sort of interested in looking at sampling strategies and sampling, not protocols. I mean, if you have unlimited funds, that's great, but we usually judge for about 40 centimeters, a rough skeleton of a core. And then if you have a lot less, like if you find that you reach this equilibrium depth sooner, then great, you can really enhance that resolution in the top part of the core. If not, then you keep going. So it really depends on the peatland, and it depends on where you are in the peatland. I think we had worked on some cores from the tropics as well, from the Amazon, the Peruvian Amazon, and I believe that also went to about 40 centimeters depth, but that was only a couple of cores.
Thank you, Nicole. I think, yeah, I had some cores also where the lead-210 goes deeper than you would expect.
Yeah.
I don't see any other questions, but I do have some, but I don't mind asking them to you a bit later if we're running out of time. If there is any other questions, Linto, I don't know if you have any more questions.
I don't see any, but one more question regarding the preparation of the sample. Now,
yes.
Normally when we do it, we homogenize to very small powder, like we go for beta or this alpha method.
Mm-hmm.
So in case of this peat, how do you do that? How do you achieve that much of homogenization?
Yeah, the homogenization, I did work on some lead-210 in different systems as well. So like estuarine samples, marine samples, riverbed samples. So in that case, homogenizing is very important because you have to do particle size analysis as well, and you have to be able to separate all the different particles. With peat, it's variable depending on the peat, but normally I just grind it. So sometimes, very rarely, but I've worked on a couple sites that had some sand as well that was blown in from the shore. And in that case, you don't really want to grind it too much because we know it's not locally deposited sand, it's been blown in. So we don't have to be as worried as with more mineral samples. Because we just throw acid onto it and all the organic material just disappears. So.
Nicole, there is a request for you to share slides. You might not be comfortable with some of the unpublished results, but maybe you could share, I don't know, Linto, if there is any way that the speakers can share the slides.
I think we have this video in the YouTube live, so they can access it. Yeah, I think that would be the best. No need to share the slide if if they have reservations.
Thank you. I was just wondering if it would be easy for you to share a link to the R package. I mean, it may be that people can find it in R easily.
Oh, of course. Yeah, I didn't even think of that. But it's if you go to Marco's page here on the GitHub, he has a link to the R package, and there's lots of information. He's made public user guides, really useful figures, like some of the figures that, a couple of the figures that I showed, and then how to use some of the different codes. So you can find everything on his GitHub page, or you can just look up on CRAN. You can look up Plum, rplum. The name of the package is rplum. Or if you're familiar with R, excuse me, you can just install it via R. So using the code install packages brackets rplum, and then use it as that way.
Linto, can I just check if we can extend a little bit, if Nicole doesn't mind, extend a little bit the time, or are we tight for time? We need to stop.
It's all right. If you have any, like a few more things to be discussed, we may take, I guess, yeah.
Thank you.
I'll just say, I'm just sharing the code for anyone who needs it, but I would recommend going to Marco's GitHub page or contact me separately. My email is on this screen as well, next to some weird yellow lines. I don't know where those came from. But there are a couple of restrictions with which versions of R need to be used. So if you have a version that's too old, it won't work.
Okay.
For example.
That's
so
great.
I'm happy to, I have a list of all of the considerations.
Thank you for sharing your email. I'm sure we will be contacting you, Nicole.
Sorry, my autocorrect keeps correcting it.
Okay, and I have a question about the uptick that you see on that exponential issue with the classical model compared to Plum. But I don't know if you could share your prior slide where you actually see some of the Plum output.
This one? No.
Yeah, for example, here, for example, in that one, or that. Well, it's actually more in the one where you can see the R.
Sure, this one.
Yeah. So there is still that sort of kick up, right? Do you think that's realistic? Is that true? I mean, if you consider, if you remove that from 22 to 26 part, then you see that radiocarbon dates have also possibilities that are much lower in age.
Yeah.
So you think of a possible alternative age-step model that passed a lot lower. How realistic do you think it is?
I do see what you mean. This case is kind of a particular one, though. It's this core is the default core for Plum, and we chose it because it's really well like the stratigraphy is really nice, and there's some interesting things happening in the peat. But then I guess for an example core, it's maybe not the best case. So we're going to add some more example cores. But this core is it's on its side, so you kind of have to turn your head. But this stratigraphy change is what's making this kick in this case. So you do have a change in stratigraphy between the whole top of the core is like really fluffy, fresh-looking sphagnum, and then just below where this change happens is really, really concentrated, like lignous material. So our hypothesis is that there was permafrost at this stage, which matches with the climate records and all the other cores I have for that site. It's all around the same time period. So in this case, this uptick would be like a slowdown in accumulation.
Yeah, you would expect. Okay, that's
and because we don't have any radiocarbon dates around that 25 centimeter depth, like ideally we would have some around this area to know how long the slower accumulation was, because within the radiocarbon date range there is quite there's a lot of options in those giant ranges. But again, it's Bayesian, so the more information you give it, the better the model is.
Okay, that's good to know. So Plum is basically suffers less from that problem from.
Oh, it doesn't at all, because it has it doesn't incorporate it incorporates radioactive decay, but it doesn't have only that. It has a lot of other parameters, like the influx, for example, that go into the equation. So it makes it a much more flexible model, whereas the CRS model, because it just has that one component, kind of always has a more or less pronounced.
And then I would like your opinion on another issue. I know you said do not use any cores for which you cannot reach the background level. But what happens, sometimes we do not have.
Yeah, I mean, I was just saying it from a modeling point of view, but you can, and we have. It's just, so for example, imagine we have this core, but we don't have the last 4 points. Because you have quite a nice decay, like a little slopey bit already, it is possible to infer a tail to the curve. And you can tell Plum to do that.
Okay.
Or you can do it for the CRS model as well. It's just, there's a lot of uncertainty with doing that, as with any HDF model when you're inferring stuff.
Yeah, I wondered if you could show the slide where you actually show how the different SCR and Plum differ with the 3 possibilities where you have all of the samples, then skipped samples, and then no samples at the end.
I had a thought while we're still on the slide, actually. I didn't show this, but what Plum can do, because I'm working in ombrotrophic peatlands, but what Plum can also do is, if you're using gamma spectrometry, for example, you will get a measure of the background automatically. So you're measuring like radium, I think 226. So Plum would have an input spot for that. So there would be more columns in this input slide where you would input the background. So the background would be like little lines on the bottom. Sorry, I didn't download an example of it, but instead of having this curve, it's like that animation that Martin Blau had produced where you see this kind of background radon. So in that case, you don't need to have this bottom because you know what the background is. So that would be one method. Or if you have other cores from the same site that do have a background, you can make the assumption, because it's Bayesian analysis, you can assume that, hey, from the same site, I assume that the background is the same.
Okay.
So you can input that as a parameter. There's these little boxes on the top are all the prior information that you modify or don't. These are all default values for now. But this parameter phi is the influx. So you could always tell the model, hey, I know from this other core that this is the background. So in that case as well, you wouldn't need that background. But you were asking about this?
Yes, there is another thing on the chat, but before that, can we just go to that slide where the 3, this. Yeah. I didn't actually, I can't see, maybe it's just my eyes, but I can't see the
I think it's quite small.
No, the difference between purple and red lines on the actual, like I only see red lines. Is the purple the really clear ones, the really dotted ones, the small
The purple lines are the dotted lines, yeah.
Okay, great. So, in the case that, because we do have cores where we don't reach the background, you say that basically Plum does slightly better job, basically?
Yeah, it's still not great, but I can maybe zoom in so you can see it better, actually. Like, is that any more clear?
Yes.
Okay.
Yeah, that's very clear. Yeah.
Yeah, so I mean, the green is the true data in this case. This is one example. So as I said, Marco's running a lot more. So maybe it will be, oh, you're doing the yellow.
I'm not.
Not
doing the yellow.
No, I don't know. Someone's doing yellow. Yeah, so you can see that, I mean, so Plum in this case is giving a wider uncertainty because it has less information. So the uncertainty includes the real data, which is a good sign.
Yeah. So would you say that the first, let's say, for example, the first part of the profile, so we would be able to use it maybe for the first part of the profile, but not for the latter part of the profile?
Well, I mean, it's a case-by-case basis. I don't really, oh my God, I'm not doing this.
What
is happening?
I don't know what
I'm not touching anything.
Me neither. I don't know
It's
what's
just going to be a child scribbling on my my slide. I think it's a case by case basis. I don't think I can answer that question offhand because it depends. Like, do you have any information at all? I think Plum would tend to be more reliable, at least in this case, because it does include the real data set. But then you can also add, if, as I said, if you have information about other cores from the same site, you can add that into Plum, whereas the CRS, you can't really do that.
Okay, and there is a note from Paul on the chat that says, do you see it? I'm using Plum and I struggle to find info on how the spreadsheet needs to be labeled for Plum to run. It doesn't seem to be in the documentation.
Okay, that's good feedback. I think I think Marco did have a sheet, like an info kind of user manual of how to set up the spreadsheet. But I think he sent it to me directly because it was hidden somewhere obscure. So, so yeah, that's a good point. I'll let him know that it is somewhere obscure, or we'll put out something sensible. But yeah, I mean, if I guess you said you've been using Plum for a little while now, so I'm sure you found how to set them up. But if you need to, these are the columns that you need, and then you can add the columns for the background for the radium 226, and you can add some other columns for like set parameters, so like the input date and how many samples you want to use to judge to estimate the background.
Okay.
But yeah, maybe another sort of user manual with like specific screenshots like or something would be helpful. Yeah.
Of the inputs, but that's fantastic. Thank you so much.
But they really like having input as well. So you're welcome to just email him. Like, he loves talking about Plum. So and they're all statisticians and modelers, so that's why they like having me around, because I'm always like, I don't know how to do this, so please help me. So anyone else who can do that, it's great.
It's been a really interesting chat, Nicole. Good luck with your recent accumulation data set. Oh, it's loads of scrubbing now. Good luck with the future work where you apply all of this.
Well, keep your eyes peeled for some parallel papers coming out at some point.
Thank you very much, Nicole. And with that, we close today's session, and actually the whole session is closed with this talk. So thank you, Nicole, for being our last speaker. And I leave Linto to say some words, maybe say thank you to everybody that has been listening as well.
Thank you, Nicole. It was a wonderful session. And I believe that we had in total 9 sessions. All were very nice, very well done. And we have got a very wide variety of like expertises, and all new proxies were discussed, many of them. So we really had a very good time. So thank you all the speakers on behalf of the organizing committee, and of course Angela and me. It was a wonderful session. It was really a good experience to listen every one of you. Thank you so much. And I hope that some of the students, maybe I've seen that some very serious interaction. So I'm sure that some of them will take up this as an area of their research. So let's have this learning experience more fruitful in that point of view. So thank you all the speakers, and thank you all the listeners. I think we will have a final word from our anchor here. From here, Ms. Asha will be saying a final note. So thank you, everyone.
Thank you, Linto. Thank you.
Hello, that brings an end to our first session, New Proxies of Climate Change of the Past and the Present. I thank all the presenters. We really had some interesting presentations. Hope you all enjoyed. And let me express my sincere gratitude to our session chairs, Dr. Irinjalakuda and Dr. Linto Alapat, who had done a wonderful job, and the session went very well. And thank you, ma'am and sir, for all your input and comments. Some instructions to the participants. We have provided a feedback link. Kindly fill it and let us know how it is going. And we will be back tomorrow with another interesting session, Geohazards Mitigation and Management. See you all tomorrow. Thank you. Bye.
Okay, thank you. Bye-bye.